Hello. Hello. Hello.
సార్ ఇంకా కనెక్ట్ అవ్వలేదా? ఓకే సార్, ఓకే. గుడ్ మార్నింగ్ అండి. ఒకసారి టిఓటి ప్రెజెంటర్స్ ఎంతమంది ఉన్నారో ఒకసారి అటెండెన్స్ చెప్పండి. ఎన్ఆర్సి వైజ్. శ్రీకాకుళం, ఉమెన్ శ్రీకాకుళం, Women Srikakulam. Pierre Kakinada.
Sir, responding, sir.
NRCs Srikakulam, sir. Morning, sir.
Good morning. చెప్పండి ఎంతమంది ప్రెజెంట్ అయ్యారు?
15 members, sir.
15 మేడం, 15
Sir.
పిఆర్ కాకినాడ.
Sir, Pierre Kakinada responding. 42 present, sir.
తణుకు.
Sir, 16 present, sir. 16.
ఓకే సార్. గుంటూరు. ఎన్ఆర్సి గుంటూరు. చిత్తూరు పివికేఎన్ చిత్తూరు. సార్ ఎంతమంది ప్రెజెంట్ అయ్యారు ఒకసారి చెప్పండి పివికేఎన్ చిత్తూరు. సార్ ఆడియో వినిపించట్లేదు మీది. సార్ క్లారిటీ లేదు, మీ ఆడియో క్లారిటీ లేదు, ఒకసారి చెక్ చేసుకోండి.
హలో,
ఉమెన్ కడప. ఉమెన్ కడప, ప్లీజ్ రెస్పాండ్.
Sir, responding, sir, women Kadapa.
మేడం ఎంతమంది ప్రెజెంట్ అయ్యారు?
Sir, 20. Sir, after seconds, sir.
ఉమెన్ కడప ఒకసారి చెప్పండి మరలా.
Women Kadapa.
మేడం ఎంతమంది ప్రెజెంట్ అయ్యారు?
30, sir, 30, sir.
13.
30.
ఓకే మేడం. పివికేఎన్ చిత్తూరు చెప్పండి. మేడం చెప్పండి ఎంతమంది? టోటల్ 27. ఓకే మేడం. ఓకే సరే మేడం. కనిపిస్తున్నారు మేడం, ఆడిబుల్. ఓకే ఓకే మేడం.
హలో సార్,
27
GCW గుంటూరు. సార్, GCW గుంటూరు సార్.
గుంటూరు చెప్పండి సార్.
22 participants సార్.
20
All are present sir.
ఓకే సార్. ఓకే
22 all are present.
ఓకే సార్.
ARP 5 members all are.
ఓకే సార్. ఓకే, థ్యాంక్ యూ. పివికేఎన్ చిత్తూరు.
Thank you.
సార్ పివికేఎన్ చిత్తూరు ఒకసారి మ్యూట్. 20నా సార్? ఓకే సార్. ఓకే, థ్యాంక్ యూ సార్. పివికేఎన్ చిత్తూరు.
Sir, clinical degree guys, sir. 20 members are attended, sir. 20 members
సార్
present.
పివికేఎన్ చిత్తూరు మ్యూట్.
20 members sir.
20నా సార్?
20, sir.
సార్ నెల్లూరు, ప్లీజ్ రెస్పాండ్.
Anantapur, sir. 23 present.
Hello, sir.
అనంతపూర్ చెప్పండి సార్, ఎంతమంది ప్రెజెంట్ అయ్యారు?
23, sir.
23. సార్ నెల్లూరు చెప్పండి.
Sir, Nellore 14 present, sir. Out of 20, like 5 allot, attending from Guntur, sir.
Okay, madam. Okay.
3 KRPs present, sir.
Okay, madam. Okay.
Good afternoon, sir. Submission, sir. Good sir.
Sir, మీరు అన్నారు.
Sir, yeah, sir. You permit us, we can do this, sir.
Very good morning, all of you. So I think resource person join here, sir. I welcome today's resource person, Dr. Professor Anish Kumar garu. Welcome, sir. Small instruction to all the participants. కొన్ని సెంటర్స్ లో అటెండెన్స్ అనేది రెగ్యులర్ గా ఉంది. I request all the program coordinators, principals and KRPs దాన్ని కొంచెం సీరియస్ గానే తీసుకోండి సార్. ఎందుకంటే కమిషనర్ చాలా దాన్ని ఏమంటారంటే ఫోకస్డ్ గా ఈ ట్రైనింగ్ పెట్టడం జరిగింది. మీరు అబ్జర్వ్ చేస్తున్నారు ఎవ్రీ సెషన్ లో కూడా క్వాలిటీ ఏమాత్రం కాంప్రమైజ్ కాకుండా ఉండడానికి డైరెక్ట్ గా కేఆర్పిస్ ఎవరైతే ట్రైనింగ్ ఇచ్చారో వాళ్ళని హ్యాండ్ హోల్డ్ చేయమని చెప్పడం జరిగింది. వాళ్ళు చాలా బిజీ పీపుల్ కూడా, బిజీయెస్ట్ పీపుల్ కూడా మనం ఎంగేజ్ చేసి ఆ క్వాలిటీ కాంప్రమైజ్ కాకూడదనే ఉద్దేశంతో పెట్టడం జరిగింది. అయినప్పుడు కూడా కొన్ని ఎన్ఆర్సీస్ లో కాస్త అటెండెన్స్ తక్కువ ఉండటం, ఉన్న వాళ్ళు కూడా అటు పెట్టడం అనేది కంప్లైంట్స్ వచ్చినాయి. ఈ విషయంలో నోటీస్ కమిషనర్ నోటీస్ లో ఉంది. కాబట్టి ఒక్కసారి ఈ రోజు నుంచి మాత్రం మనం చాలా స్ట్రిక్ట్ గా ఫాలో అవుదాం. డ్యూరింగ్ ది సెషన్స్ ఎవరు కూడా డిస్టర్బెన్స్ లేకుండా కూర్చోవాలి. ఇది చాలా యూస్ఫుల్. అంతేకాకుండా అవుట్కమ్ బేస్డ్ అనేది మనకి కమిషనర్ గారు ఏం చెప్తున్నారంటే ఈ సెషన్స్ అన్ని అయిపోయిన తర్వాత అందరూ కూడా ప్రాజెక్ట్స్ ఇవ్వడానికి కూడా ప్లాన్ చేస్తున్నారు. మీరు సబ్మిట్ చేసినట్లయితే దాన్ని దృష్టిలో పెట్టుకుని ఇప్పుడు ఎవరైతే మీకు ఎవ్రీడే క్లాస్ చెప్తున్నారో వాళ్ళ యొక్క కొలాబరేషన్ తో మనం రీసెర్చ్ అనే దాంట్లో కూడా ముందుకు వెళ్తాం. కాబట్టి దీన్ని సీరియస్ గా తీసుకోవాలని మనం థ్యాంక్ యూ మచ్. థ్యాంక్ యూ సార్. యూ కెన్ ప్రొసీడ్.
Thank you.
Very good morning, dear participants. I am Vasanthi Rashmi from NRC Kakinada. I take the privilege of introducing today's resource person, Dr. P. Anil Kumar garu, who is working as Associate Professor of Biochemistry at the University of Hyderabad. Sir is an eminent personality. He has number of achievements, awards to his credit. Sir completed his graduation in microbiology from Nodal University, Guntur. He has done his masters from Andhra University, Visakhapatnam during 2002. In 2008, he completed his PhD in biochemistry from NIM ICMR, Usmania University, Hyderabad. From 2008 to 2011, he was postdoctoral fellow, University of Michigan, USA. So this is his academic background. From 2007 to 2012, 2007 to 2008, Sir worked as postdoctoral fellow at University of California, USA. From 2008 to 2012, PDF from University of Michigan, USA. From 2012 to 2014, Sir worked as DSP Inspired Faculty, National Institute of Nutrition, India. From 2014 to 2024, he worked as Assistant Professor, Department of Biochemistry, University of Hyderabad. Since February 2024, he is working as Associate Professor, Department of Biochemistry, University of Hyderabad. He has number of awards and recognitions. He received Research Fellowship Award by CSIR India and ICMR during 2002. KV Rao Scientific Annual Research Award, Hyderabad during 2007. Endocrine Trainee and Travel Award San Diego CA during 2010. DST Inspired Faculty Fellowship Award during 2011. DBT Welcome Trust Early Career Awardee during 2012, but he has not availed that award. FA SEB Travel Award Snowman's Village, USA during 2012. Wood Whelan Research Fellowship during 2014. ERN EDTA Travel Award to participate International Polo Site Conference, Israel during 2016. DHR India Long-Term Fellowship to visit University of Michigan during 2018. Sir received Canada Gold Medal from Bionest University of Hyderabad during 2019. Sir is an elected fellow of Telangana Academy of Sciences during 2021. He received the Chancellor Award at University of Hyderabad in 2022. He received Young Scientist Award Education Expo New Delhi during 2022. He received ICMR long-term fellowship to visit Harvard during 2024. So these are some of the awards and recognitions of Dr. Pasupuleti Anupkumar sir. Coming to is the research projects. Sir completed 11 research projects. Funding from three with ICMR funding, 2 with the DST, 3 with the SEP, 1 with MSRP, 1 with CSIR, 1 with NRC funding. Sir guided 6 PhDs, he 20 MSc dissertations. He is the author of 5 books. He published 65 papers in renowned international journals. His research areas are, he is interested in diabetic nephropathy, chronic kidney disease, epithelial mesenchymal transition, and bioinformatics of kidney. So with this brief introduction, I welcome Dr. P. Anil Kumar, Associate Professor of Biochemistry, to enlighten us on the prevention of post-defense mechanism. So, sir, we welcome you, sir. The floor is open for you, sir. You can continue with your lecture. Thank you so much for this opportunity.
Yeah, I thank the Andhra Pradesh Higher Education for giving me another opportunity to interact with you. I hope you people can see my screen, my slides. Okay, great. Thank you. Actually, I'm afraid of having an elaborate introduction about me, but that's slightly enough, afraid. Anyhow, because the most materialistic things are, with us is the most important thing is, getting our conversation about host defense mechanisms. See, if you ask a question to yourself, faculty, you might be teaching immunology, you might be knowing what is antibody, you might be knowing what is antigen, what are antigen antibody reactions, allergy. The simplest thing from to complex thing, you might be knowing it. But to begin with one thing, I think if at all any video audio connection is nearby, just you can say answer. Otherwise, also fine for me. The simplest thing where you will use your immunology background in common life, simplest thing. Oh, you don't have audio, right, with you? I mean, you can't talk to me, right? Or you can say, I'm afraid. Madam, you can say some words or just you have to passively listening to me.
Yes, sir, we can.
Oh, you can? Okay. Oh, I mean, when it is Pradeep, Dr. Pradeep, this participants also can say hi to me. I mean,
Yes, sir. Yes, sir.
Oh,
They will
great.
come to near and there.
Acha, wonderful. Actually, I'm trying to. Unfortunately, I cannot know your names. Sorry, but the simplest thing what you come across, like immunology, normal life. Can you say something like, what you are? If someone will ask you what is your blood group, how do you know your blood group? You have to go do some small immunology test, right? You might be A, B, A positive, B positive, O negative, AB positive, AB negative, right? So from simplest thing in common life to advanced about corona, we all worried about corona. Oh my God, what is this one? How to get immunity against that one? Fortunately, with our background, many of us are survived because luckily the disease did not affected much of us. The reason was, fortunately, we people are, have some immunology, some immunity background for the very disease like that. So we have so much of the information about immunology. Being a faculty, you might be knowing it. But for next 1 hour, we'll try our best to talk about some evolutionary aspects of immunology. You able to hear my voice properly, everyone? Dr. Pradeep, my voice is clear for you? Or you want me to loud?
Yes, sir.
Clear.
Okay. So I request all of you, just imagine a scenario, for example, in a society where in New York, 76,000 people passed away during Corona. 76,000 people in one city in the span of 5 months. 5 months. Okay. First month itself, 25,000 people passed away, and the remaining 4 months, remaining 50,000 people passed away. See, if just how evolution for the host defense mechanism will evolve. I would like to request you people, just think something like that. In New York population equal to Hyderabad population, right? Like almost a crore population. But 75,000 people died very in a quick time, but remaining people are survived. What happening here when people and pathogens, human beings versus virus, host versus pathogen, synonymous, host or guest or organism or what do you call Homo sapiens or human beings. Other side, virus, bacteria, pathogen, this kind of microorganisms, one side. Now, what really happens when we both come in contact, just come in contact? See, do you think all of us are infected by Corona in the first infection, first wave or second wave? Is it true? Everyone is infected in the first case. So now you could see here a simple thing. Don't worry about so much of the drama on my slide. Just go with first one. You could see when we and pathogen, host and microbe come in contact, what you call it as infection, right? Infection means they may come in touch with our skin, they may get into our nostrils, or they may go into our body through water. If any wound is there, they get into this one. So this is called infection. It's not called disease. It is just called infection. Sometimes you feel some little throat for half an hour or 1 hour. If you have some hot tea, ginger tea, lemon tea, it's gone. Or you will take some strepsils or some Vicks tablet, it's gone. Its initial thing is infection. So once you got infection, you can eliminate it very easily because you have very protective effect. Like now, it is called elimination. The first thing that is going to happen is elimination, immediate. So second thing, what happened, without our knowledge, even though bacteria comes into our body or does not know in our body, we don't know that also. It will just enter into our foreign or enter into our body. It can become a commensal. The commensal means, anybody can say what is commensal? Anybody who has access to computer, can you say what is commensalism? Commensal means? Hello?
An organism is benefited, sir.
Oh, very good, madam. Very good. Thank you. I think the people from Kakinada, they said this one. Thank you so much. Excellent, madam.
Sorry for the inconvenience. Please maximize your screen, sir.
Okay. Now?
Now it's better, sir. Thank you.
Okay. Please ask me. Okay, great, madam. So whoever said the answer, and thanks for bringing me that I have to upgrade screen. Okay, great. Someone said answer to comments. So pathogen is getting into our body. It is getting benefited, or we are getting benefited, madam? Without our knowledge, that is getting benefited, right? It is getting shelter. It's got a room to stay forever, whatever time. Slowly, after staying for some point of time, this commensal is trying to help with the host. So now host and pathogen both are getting benefit from each other. The condition is called as
Symbiosis.
Very good. So this both commensal and symbiosis does not notice to us like infection, like disease. We don't know. You know what is the perfect example for symbiosis, madam? Anybody can? What is the perfect example? Don't tell me rhizobium.
Lichens.
Huh?
Lichen, sir. Algae and
Lichen.
fungal association.
Excellent. In our body, madam. In our body, madam.
White termites, Plasmodium. White termites, equally.
Symbiotic bacteria.
Equally.
Perfect example. Mitochondria, chloroplast, right? Yes or no? They're not actually the part of our cell. The blue-green algae came into some other eukaryotic cell. It become chloroplast. Same thing, mitochondria. That is why both are now helping each other. Mitochondria helps with energy and to the host cell, and host cell provides the necessary things for mitochondria. See, madam, just imagine, go back to 1 trillion years ago, see that what is exactly happening in it. Individual cells are there. A small cell encompass big cell. When the big cell don't like the small cell, it will chew it contents and break down into small molecules. When big cell is little concerned, little kind enough, the small cell which get into the big cell will stay for some time. Settle in the cells. That is called commensalism. Slowly, they both talk each other. Okay, give and take, quid pro quo. Naaki business help chayi, nenika business help chestanu. That is how they got into symbiotic relationship. Okay, that is but symbiosis and commensalism, they does not notify to you because it will evolve over a period of time. It doesn't hurt you. It is also a host interaction, but you cannot call it as a pathogen. Just it's a microbe gets into the host. This is how we all evolved. One person said lichens, fungal and algae, very excellent example how nature has come. Because if lichens not have formed, probably plant would have not evolved. Okay, man. Let's keep aside that one. Now we'll go to the first one, elimination. Second one is B. Madam, if everyone please try to see a situation where how we people talk each other. First infection comes, it will eliminate it, so you will be healthy. But second thing, what happens in corona? Many people got escaped. Just pathogen is eliminated from them because you people might be very young. Second, we are black race. That means people in the black race got somewhat immunity against some, especially viruses. The third most important thing, we people live in, sorry to say, slightly unhygienic atmosphere. So we might be already exposed to similar pathogen. That's why we are escaped. But among us, some people got infection, they died. Especially suppose, for example, the famous personality in Telugu states who passed away during corona is SP Balasubramaniam. He didn't do anything. Just he went to some place. He had a mic used by somebody who had infection, and he got so close. He went right so close because his nose and mouth are so close. He passed away. So that means second thing, people get some disease. After the disease, they'll eliminate. He was fighting in a hospital for almost 10-15 days. He has undergone ECMO, ventilator, and all, death. You see the problem here. Host and pathogen come interaction. How we evolve. 80% people in India, they got infection, they survived. 20% people, they got infection, they went to so critical stage and passed away. Right? You could see now after infection, there is a B. There's a big line, is there a B? So then the big line, it is called disease manifestation. A is pathogen got eliminated. Pathogen will die in our body. So you don't even see much infection, just very healthy. Second one, the interaction, host and pathogen interaction, tug of war, fighting. Slowly, pathogen is overtake our body. It causes disease. Okay, once disease occurs, naturally there are two things will happen. If bacteria want to win, pathogen want to win, virus want to win, we'll die. The second thing, what is the problem is, if they die, then we'll be eliminated from the infection. So once we eliminate from the infection, which is called as a disease comes and we got protected. For example, you could see there are something called immune and therapeutic eradication. You see the letter G. You could see letter G somewhere. Anyone? You could see now if you write in your notebooks. You could see in the notebook, sorry, you could see in your somewhere where, please write this small word, madam. Why I'm telling this one, this small tiny letter, what you want to write. A, then B. After B, there are two possibilities are there. Number one possibility is that when we get disease, the bacteria will be therapeutic eradication. We can eradicate the bacteria or virus. That is called G. If that is overtake us, ultimately we will be eradicated. That means called death. Right? But there is one thing I want to tell you. After disease happens, sometimes HIV infection, what happens? If a person gets HIV, our cells trying to combat one, and bacteria, HIV virus is so smart. HIV virus gets into some of our cells and stay hiding there. That is called latency. Okay, that is called latency. So after 10 years of a latency, what happened to this pathogen? This pathogen will be overtake sometime later and cause disease. That is why HIV people, first 2, 3, 4, 5, 6 months, they are positive, HIV positive. After that, they will be HIV negative. Once their immunity goes down or they become aging, again HIV positive. So that is the reason why they will be causing that disease manifestation. So I request you people, this is actually research guidance. I want you people make a small note of it, this whole diagram. So now what happens? Disease comes, they may go latency, again disease will come. Throughout our lifetime, we have to constantly battle with the viruses or bacteria, anything that comes in our body. That is why there is a 24/7 battle. Another thing I want to tell you, interesting thing. Once we come in contact with the pathogen, it is tiny bacterial population is not a colony. This colony, till it becomes so many mature, quorum sensing. For example, you see some people attacks us. If you're more people, they hide somewhere, they increase their number. Once they got their number, they come back and attack on the main thing. For example, you could see what happened in Jerusalem. The Jerusalem people, there are very few people are Jews. They waited for some time. I think just to bear with. Excuse me. If you could see this colonization, what is a colonization? Means very important word, quorum sensing. That means suppose your enemy is so big, suppose our host is so big host, what the bacteria does here or virus does, they try to increase the population. So these are all evolutionary tricks for them, right? So colonization will come. Even though the colonization, they increase the number of the population, what is going to happen here is that after colonization, they try attack us. They cause either disease directly, that means E, you see the E letter there, or else they undergo latency, that is called F. Alternative to this one, elimination. Even though number, but host is very strong. The strongness of the host depend upon 4 things. I'll repeat for you. The strength of the host depend on 4 things. Number 1 is your prior exposure. Prior exposure means similar kind of bacteria might have infected other virus might infect. We had some kind of immunity. It's called memory of the beta cells, memory of the lymphocytes, one of the lymphocyte cells. The another one is, most crucial is your nutritional effect, our aging. Third most important thing is how your body will be respond to the immune system. See, there is a saying in Telugu, what you called godito poyedani goddal endukani. Am I right? And చిన్న పామును కూడా పెద్ద కర్రతో చంపాలని. There are two mutually contracts, right? మీరు చెప్తారు ఆలస్యం అమృతం, ఆలస్యం విషం, ఆలస్యం అమృతం విషం. Same way, how you respond. Just for example, something happened immediately next to you. You saw a snake next to you. If you balanced, suddenly what you do, you slowly escape from there. Or you try to put some object between you and that snake. If not, you jumped on somewhere, slipped on from the chair and fell down. Will also got injured to our head. Same way, overreactivity. Suppose human body has a tendency to overreact. This is called hyper response. And also what is it called? Storm, cytokine storm. Storm means huge thing. Most of the deaths because of Just for example, I will tell corona is a good example. The deaths in corona are happening because of cytokine storm. That means for small levels of the virus, our body is reacting more. So this hyper reaction is not good. Okay, so many factors influencing. So you could see this web of the host pathogen interaction. Mainly two things you have to notice it. One, infection occurs. In case it may cause a disease. From disease manufacturing, again, four possibilities are there. Virus or pathogen will die, or we will die, or pathogen undergo latency, and latent pathogen again come and back cause the disease. This is most one thing. The second one, colonization. The pathogen enter colonization, slowly it will wait for the situation, it again causes disease. For example, UTI infections are there for women. These UTI infections are, some pathogen will come in for very small amount in our body. Our good pathogen, good bacteria in our body fighting against that. But whenever you are low in your esteem, not healthy, other factors are playing a role, suddenly this pathogen is going there. And that's why we cause UTI infections. Over a period of time, again, what you call our body immune system will cope up, it will eliminate the pathogen, or else you may take some medicines, it will also protect you. Okay, but taking medicine is not a evolution. I'll repeat for you. Taking medicines is not evolution. We are not evolving anything by taking medicines. We're only relieving some symptoms. In order to make the better future, I'll tell you how bacteria will evolve. Without bacteria won't take medicines, right? If your animals are there, take any animal, your dogs, cattle, or pet animals, cheetah, wild animals, elephant, they never go for medicine. They don't have a doctor, they don't have RMP, they don't have a multi-special hospital, but they naturally they evolve. You see the evolution. Bacteria is infected by viruses. We call bacteriophages. This bacteria can infect fungus. Fungus infect higher organisms, right? Every step there is something. If you see even bigger organisms like crocodile lives in such a dirty atmosphere, it does not get any disease. Honestly, it does not get any disease because of the unhealthiness or uncleanness. I'll tell how it happens. Okay, now you could see these are the common words. Don't worry about this. Resistance means ability to ward off a disease. Majority of us are resistant to at this point of time because we are very middle-aged or very young. Some of you were very young. And this varies among the individuals within the same species because some are very nutritious, some are eat very raw food, some are eat very good food, I mean quality food, not the obesity diet, but very good food, very good protein diet. Some eat more carbohydrates. People who eat more carbohydrates, they're less immune than people who eat more protein. I'm repeating for you. People who eat more protein, they're more immune than people who eat more lipids because lipids cause inflammatory situations. And immunity is a mechanism of used by the body as protection against microbes and other foreign agents. Non-specific immunity. This is non-specific is given by mother through milk. When we born, some kind of placental transfer it will come. And specific immunity is against the pathogen. Most of the immunity what we get is against the specific immune. Like you suppose once you get malaria, we never get malaria. Once you got chicken pox, measles, we never get a disease. Once you got diphtheria, Corinth dagu, we never get disease. These are all. Once we get polio, we never get again. These are called. Once we get lifetime, until we die, immunity stays with us. Now I want to show important evolution. Just in the terms of evolution, I would like to show something here. If you can go through this entire slide, this makes most meaningful thing. This is not there in any textbooks because this is a research what we should. I want to ask you, do you have any pet animal in your house? Anybody has a dog, pet animal mainly? The pet animal lick you, pet animal uses its saliva to touch you. So nobody is talking and everybody is silent. Hello? Anyone, sir and madams? Do you have pet animal?
Sir, yes, sir.
Okay. So what does this pet animal does actually? Second thing, I know most of you, maybe any one of you from village background or rural background,
Yes,
in your
sir.
childhood, when
Yes,
you're
sir,
playing
yes,
in between playing, you might have got thirsty and you never go home, but you just go to pond and go to the pond and do like this on the surface of the water and take the water and drink it. How many of people drank the water like that? And you're perfectly fine. Nowadays, you need Bisleri bottle or some kind of aquafina. Otherwise, you will feel very bad. You can't drink the same water again now. Why? What's happening? So sterility, right? Your body, this cavity has a lot of protection. Your water is same now. Maybe now little dirty is fine or maybe clean. See, we have saliva, right? Saliva has a lysozyme. We have ice. What do you call it? When we have a little, when we're crying, little water will come. It have a lysozyme. Our skin, especially women's skin, is little softer than men because they have more sebaceous gland. The sebaceous glands gives what you call the low pH, and bacteria will die there, right? Or fungi cannot grow there. These are the protection. These are evolution of us, right? Especially, can anyone tell the answer? What is the mechanism of action of lysozyme? How lysozyme protect us? So you cannot be silent. There will be you will have, if you tell the answer, there's a lot of appreciation by your colleague. How lysozyme protect us from pathogens, bacteria?
Sir, that is on the cell wall of the foreign materials.
What is cell wall is made up of what, sir?
They are made up of proteins and
Peptidoglycan, sir.
Very good, madam. Peptidoglycan. Sir, please tell me, madam, I think you please have this vision, madam, so that when you people are from NRC Guntur or NRC Kakinada, you just don't remove that video. Just you won't talk also fine. Sometimes you talk, that's good. If I see the people, it is like connection between you people and me. A lot of Kakinada, please keep that one, and also Guntur. It's a very good answer, madam. Peptidoglycan. What peptidoglycan made up of what, madam? What are the bricks in peptidoglycan wall? There are two types of bricks are there.
It is NAM and NAG.
Very
NS
good.
style
What lysozyme does?
muramic acid.
Between NAG and NAM, what lysozyme does?
It breaks the
It
breaks the
Very good, madam. Lysosome break the cell wall. Lysosome break the cell wall. That's how it protects you. Lysosome is secreted by a number of places. Lysosome is there in our tears. Lysosome there is in our saliva. Lysosome is secreted by many of our mucous cells, like throughout our lysosome. That is how we get protection. Now you see crocodile also get protection like this. Bacteria will be there on the dirty mud. It will be died by lysozymes. That is the first layer of protection. And also, frog skin. The frog skin has antimicrobial peptides. The skin of the frog, it will produce a lot of, even our skins also produce a lot of antimicrobial peptides. That is how bacteria will die. Madam, in evolution, people don't have Mysore sandal soap, Santur soap, Rexona soap. Still our ancestors are lived happily. Ancestors were enjoyed the weather, and the ancestors have lived, that's what we got progeny. So there is no requirement of soaps as such, because our bacteria will be taken care by the antimicrobial peptides on our skin. I was just talking to people who have pet animal. Pet animal has a salivary IgA. What did I say? Salivary IgA. Because their saliva will have IgA. Mother kisses her baby when it's a small child. So a lot of IgA will pass through from mother to the baby. Okay, that is how it protection. Okay, this is the main thing. Immunoglobulin A is one of the molecules. See, think about this, so many evolution of the people keep on evolving. I'll tell one side example. You could see all of your, gazetted officers, first class employees. Our income is quite good. Our cleanliness is good. We'll buy houses in a very decent places, civilized places. But in the same society or nearby society, people are living in unhygienic conditions are there, who are like, what do you call orphans, or the children with, women with a low income. They're growing their children. But why their children are also very healthy? Just only sometimes they may be malnutrition because of they cannot afford good food, but otherwise they're healthy, no infections for them. Why? The reason is that in their body, when mother is carrying the baby, believe it or not, mother when carrying the baby in this kind of in unhygienic conditions or slum areas, the mother's blood will have antibody against these toxins, tetanus toxins and all. So mother will have antibody. So mother transfers this antibody to the baby through the placenta. So the antibody that passes through the placenta is dash. You people have to answer later. Some antibody will transfer from mother to the baby. That is called passive immunity. And these babies, when they're born, they're having protection against the pathogen that is already there in the society. in the dirty conditions, whatever the antibody and what pathogens are there, for them, mother has developed antibodies. It is transfer the antibodies to the baby. That is how baby gets protection. And also most of the women here, you would have noticed that when baby born, first, second, third, 4th month, baby is perfectly fine. So only after 6th or 7th months later, they will get some kind of, respiratory congestion or something. So as soon as baby born, baby is very healthy, very happy. They won't get any infection. Maybe low weight or higher weight, that is different. But that means mother, evolution is taking care of the baby. You and me also fine. Right? That is what I would like to tell you. So second thing, what is body level? Just come into the little body. Let me know inside of the body. We have so many cytokines. Yes, please. Anybody has something? I heard some whisper. You want to ask any questions? Pardon? Okay. And second thing, what I would like to tell you is body level cytokines. See, cytokines, there are chemical communication molecules between your own cells. I also said some small thing called cytokine storm. Okay, that means play in a safe game. So whenever pathogen comes into our body, we'll try to fight with them. Our cytokines helps pull down the they act as a pro-inflammatory and anti-inflammatory. Anti means they're good, pro means they're bad, and the balance is always maintained. Sometimes because of infections, pro upregulates pro-inflammatory cytokines, then we got disease. Sometimes autoimmune disease is also same thing. However, our inflammatory mediators are there, like prostaglandins, right? They help in the construction and dilation of the blood vessels. These are the things that play very important role. NF-kappa-B, this play a role. NF-kappa-B both cell level and body level. Most interesting thing, most of you taught your students in immunology, you studied about this called specialized systems, third line of defense. This is where you call it. Third one you call it immunology. In the books of immunology talk about the third system called specialized system, which is there in the slide green color. You could see from the thymus gland. You know very well about this immune systems. Thymus, there are two kinds of cells, T cells and B cells. T cells which are matured in the thymus, B cells matured in the bone marrow. And these cells will produce antibodies, and also from the memory cells, antibody produce plasma cells. Now, the blood, I will try to quickly recap this one for you very fast, so that you people can get this one. Okay, you could see this is the blood. This is actually what I'm talking about, these blood cells. These blood cells are like, they are granulocytes. And the granulocytes are basophils, neutrophils, eosinophils. And they are called monocytes and lymphocytes. Monocytes are granulocytes and they become macrophages. These macrophages are sitting there in your lungs. That is how you got protection. Any pathogen entering into your respiration, it will die because of alveolar macrophages. So then, and lymphocytes, there are two lymphocytes, T cells and B cells. Now these T cells and B cells, what happens is, these cells, they try to, because of interest of the time, I'm just moving to back and forth. I just want to tell you here is that, so these cells, lymphocytes, which are coming from there, T cells, they try to see the antigens comes into your body and present it to the B cell. B cell produce antibody. For example, you are the police. This police is a T cell. It will identify the thief, but won't punish it. It will show it to judge. Judicial system will take the punishment and jail. And jails and judicial system will take care of it, punishment. So B cell is the one that is actually producing antibodies. That's how, and there's a complement system, very ancient system. It will try to penetrate the cell walls of the bacteria. These are the things actually which are evolved. Now you people will appreciate actually our evolution of the system. Very, very crucial thing. Have you ever heard of the cellular level immune system, like RNA interference, CRISPR-Cas? Anybody in our class, anybody heard about RNA interference and CRISPR-Cas? Especially women faculty should know, because CRISPR-Cas is discovered by 2 women faculty and they got Nobel Prize. And both RNA interference also got Nobel Prize. Okay. Can I show you some small video? How evolution started at a cellular level. See, difference should start at cell level, because unicellular organisms have formed first. Then we become multicellular, then full organ. Now there are so many systems are there in the pink color one. Predominantly autophagy, interferons, RNA interference, RNAL, reactive oxygen species is called ROS. antimicrobial peptides, apoptosis, P53 and cellular level. Can I show a small video for you, please? Can you see my screen now? Okay, you have to tell me what it exactly about. I'm playing small video for you. Its video is just 2 minutes. Please see this video.
RNA interference is a mechanism of silencing gene expression after the gene has been transcribed. This interference is due to the presence of antisense RNA.
Sir, video is paused, sir. You are in mute, sir. You are.
Sir, excuse me, sir. You are in mute.
Mute, sir. Just unmute.
Someone want to ask me any question because I heard some voice. That's what. Okay, you can able to hear this audio, right? Just 1 more minute, please. RNA has 2 components. I'm just going back and telling. Please listen this one.
As the synthesis.
Sir, video is not playing,
Anil,
sir.
sir.
Anil, sir, video is not playing.
Anil, sir.
చెప్పండి మేడం. హలో
Video.
Sir, video mute అయింది, sir.
RISC. The messenger RNA is then degraded. The presence of this rapid degradation
I hope you got it. See, this is actually one of the primitive evolutionary mechanism how a cell can protect herself against a pathogen. Okay, see the beauty. See, what is the aspiration of a person, right? Want to live, right? No matter what, you are poor or big or richer, aspiration of a person is to live. Same way, aspiration of a cell is to live. It does not surrender to the pathogens, right? So what exactly happening in such a small, tiny bacteria has evolved such a vast, intelligent mechanism to survive. See how they evolved it. Whenever a pathogen comes into it, it will try to recognize some small region in that pathogen's DNA, especially DNA gives RNA. When this pathogen's RNA component will bind with the host small RNA component bind to the pathogen small RNA. That's why that means whenever system recognizes double strand RNA, it will degrade it. For example, you got some new thing like new sequence. What does it do? It will try to remember the sequence what has come now and will try to pass this information to the baby cells. Because see, when a bacteria dividing into 2 halves, small one, it will get the mother cells components. So that's a smart thing, right? The way you are dividing your property and giving it to your children, the bacteria gives information of its pathogens which are infected us when it is healthy, it is baby cells. Because mother won't live once baby cells come, right? Unlike in human beings, bacteria, what does bacteria do? It will divide in the middle. Then it becomes 2 daughter cells. Okay, information is given. See, such a small evolution thing. Now, anybody, I want to ask a question. What is meaning of RNAi? What it is called is RNAi? Hello? Why it is called RNA interference? Okay. Just because it is, if it is possible two-way, then it makes sense. Okay. So this is actually most crucial thing in the evolution of system. Now I want to tell you one thing. Human brains are so smart. We are very good in copy. Like we never discover any softwares, but any software come in the market, the Telugu people can learn it overnight. The same way, human beings, we cannot generate this kind of intelligent evolutionary mechanism, but human brains are very smart in adapting them or hijacking them. Humans are using RNA interference for genome editing. For example, you want to remove a gene in tomato, use RNA interference. You want to remove the what you call brinjals, you want to improve the brinjal quality, you can use RNA interference. So that's we can imitate the nature, but we are not real creators of the technology, right? That's what great thing about RNA interference. Okay, any questions so far? Any questions? Okay, okay. So you want to see small video about CRISPR-Cas too? Hello? You want to small video about CRISPR-Cas? Do you want or no? Okay. See, because science lo kotta vaatiki, sahityamlo paada vaatiki pradhanata ekkuva. Since we are all our science faculty background, so let's see the new one thing, CRISPR-Cas. See, CRISPR, 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 CAS. CAS is a protein, CRISPR is a phenomena. I would like to play this video for you people so that at least few of you will get and excited about this new. This is a very, very, very ancient technology. It is there in evolution maybe 3 million years ago, but it was discovered by 2 women scientists 10 years ago. It's called CRISPR-Cas. Let us look at this video, please, once again, so that we will move to the next class. Next topic of the class. Please listen this one. CRISPR, CRISPR. CRISPR means, SPR means short palindromic repeats. Short palindromic repeats is called SPR. Okay, they're interspersed. That means interspersed, like cluster of interspaced short palindrome repeats. Please listen this talk. It is just for 4 minutes, then I'll wrap it up.
Molecular biology, CRISPR was first identified in E. coli. And if we break apart the acronym, it stands for Clustered Regularly Interspaced Short Palindromic Repeats. Now that's a mouthful, but it does tell you the two main parts found in CRISPR. First of all, we have the repeats. These are going to be short segments of DNA, so 20 to 40 letters in length, and they're going to be palindromes. Remember, a palindrome is a sequence of letters that read the same left to right, like never, odd, or even. So we're going to have these
Anil sir.
సార్ మీరు మ్యూట్ చేస్తున్నా వాయిస్ మ్యూట్ అయిపోతుంది సార్. ఆడియో ఇస్ నాట్ వర్కింగ్ సార్.
Sir, Anil sir, the video pointer is not coming, sir. You please unmute, sir. Sir, please unmute.
Anil sir.
Sir, Anil sir. Sir, am I audible, sir? Please, sir.
injects its DNA.
Madam, everyone,
Excuse me,
please
sir.
see this
Anil
whole
sir.
concept. Kindly please pay attention, because anyhow, a few more minutes. What I want to tell you, this is actually, we use routinely this CRISPR-Cas RNA system in the labs. Hello, please refrain from background noise, because you are disturbing both the coherence of the class. Please see, if you have a bacteria, for example, take E. coli. Generally, E. coli will be infected by bacteriophages, like any bacteria. This E. coli or other lower bacteria, they have this CRISPR-Cas. CRISPR-Cas nothing but in the DNA sequence of them, in between regular at interspace, there will be short stretch of the DNA will be there. So this DNA is palindromic. You know what is a palindromic? No, short repeats, palindromic. There is called cluster of regularly interspaced short palindromics. They will be there. You know what are those interspaced regions? They are the hereditary property. This E. coli got them from their parent. Their parents got from their parents. Like you got the properties in your lands, assets from your parents and grandparents. Same way, the present bacteria E. coli got them from the parents. What they means it? These short interspaced sequences, they mimic the viral genome. Once any virus comes into this E. coli, see what is going to happen here.
Normally would happen if you don't have an immune system is this DNA would hijack the cell. It could become embedded in the genome, but more importantly, it would make a bunch of these bacteriophages and eventually kill the cell. But since it has this CRISPR system, what it's going to do is it's going to transcribe and translate proteins, so this Cas complex, and it's also going to transcribe that DNA to make what's called CRISPR RNA, and it'll fit right into this protein like this. What is this? It's a way to fight that viral DNA. It essentially breaks it apart. And so before the infection starts, the infection essentially has ended.
See, what is saying? Before infection starts, it is ended. That means pathogen, bacteriophage, virus gets touch into them. Once touch into them, it will try to release its DNA into the cell. That's how bacteriophage release its contents, and bacteriophage will transcribe many of its copies. Once they form, they assemble as a virus, a bacteriophage, and break open the E. coli. That is called disease, right? So E. coli will die. But what happens here when CRISPR-Cas is there? Whenever the pathogen comes into this cell, Cas genes of the host bacteria are transcribed into a protein called this Cas protein. Then the target genome, that RNA will be transcribed. Now what happens? You see this once you see this one. See now the viral genome comes into interaction with the glue
complex, and it's also going to transcribe that DNA to make what's called CRISPR RNA, and it'll fit right into this protein like this. What is this? It's a way to fight that viral DNA. It essentially breaks it apart. And
see, what is the beauty of this system? In both, what is the common in RNA and CRISPR-Cas? Anybody can say what is the common in RNA and CRISPR-Cas? Anyone can say. Madam, if anyone can say, I will send this book to you by post. I'll send this notebook, which is from EMBO, European Medical Molecular Biology. I'll send this one by post if you can tell the answer what is common between RNA and CRISPR-Cas. Anyone in our faculties? This is
the short palindromic repeat, sir.
No. That is all unique to CRISPR. What is the common in CRISPR-Cas and RNA in the execution of the action?
Silencing of gene expression.
Pardon?
Silencing of gene expression.
No. Both cases, bacteria will die. I mean, gene will be cleaved. But what is the common thing, madam?
Nuclease activity, sir. That is the digestion of RNA.
Oh, very good. How, sir? Why RNA is selectively digested, sir?
That the RNA
Go ahead, sir. Sir, what is your name, sir? Sir, what is your name?
Sir, my name is Dr. Jayappa, sir, from Anantapur.
Oh, I got it. I got you.
Yes, sir. Thank you.
So no, not done. Why it is RNA selectively cleaved, sir?
Sir?
No, why is this RNA selectively cleaved, sir? Why RNA selectively cleaved? Madam, you are saying something. Another madam.
That dicer and risks have been formed now.
No, that is good. No. You are telling who cleaves the RNA. Why RNA is getting cleaved, sir? Why not other RNA is getting cleaved? Why selectively this particular case?
Sir, actually in the RNA.
Madam.
Arts College Rajahmundry.
No, madam, I couldn't hear you. Please finish it, ma'am.
Sir, myself K. from Arts College Rajahmundry, sir.
I think your voice is not very. Sir, Jayappa, sir, what madam is saying, sir? Why RNA is degraded?
Sir, in RNA, sir, is my voice audible? Is it clear?
It is cut.
Sir.
Genome
matches with the guide RNA sequence.
So, what is the same between
Sir, two different mechanisms, sir. RNAi and CRISPR-Cas, these are primitive evolutionary defense mechanisms. What is the common between them? As one of your faculty said, in both cases, RNA will be degraded. Why RNA will be degraded? That's what my question. Sir, what is the fundamental difference between RNA and DNA, sir? Anybody? What is the fundamental difference between RNA and DNA?
so RNA is a expression part of the DNA.
No, structure wise.
Sir, RNA is a standard one and it consists of a uracil in the place of thymine.
No, don't go to the detail. Don't go to the detail. Structure wise. No, don't see, sir.
Only
Sir, only one thing, sir. Evolution is so smart, sir. Every cell will think, sir. Even if it's a bacteria, if you put more salt in one direction, the B. coli will move away from the salt, right? See the beauty, sir. In the whole evolution, always RNA is single stranded. Whenever you could see RNA double stranded, system has to cleave it. That is the Cas9 or Risk and Dicer complex. Dicer, it will dice it. In the Risk complex, Cas9 will cleave it. In both cases, what happening? A pathogen when coming to your body, this host cell, sir, this much tiny bacteria is so intelligent, it is trying to make a whole pathogen's RNA double stranded. Once it becomes double stranded, Cas9 in case of CRISPR-Cas, or in case of Dicer and Risk complex, in case of RNA, they cleave it. The common between them is degradation of RNA. The further, very good, sir. But more insightful is the common between them is degradation of double-stranded RNA. That is how nature has selected that. Okay. You may ask a question again. Why DNA is a genetic material? Why not RNA? If RNA is a genetic material, you will have more infections, madam. You have more problems RNA genetic material because pathogens come and deroot it our RNA. Our RNA differentiate. The system cannot differentiate between which is RNA from the host, which is RNA from the pathogen, right? You think I gave enough explanation for you? Hello? Hello?
Sir, this is from NRC Kadapa women.
Okay, madam. Any question more for you?
No, sir. In the case of ribose, we have hydroxyl group at second carbon.
Yes.
It interferes the stability of the double-stranded RNA.
Got you, madam. You also said uracil. Time and difference. Got you. I heard, madam. Thank you. Thank you. I just, you
before the
just
infection
listen for one more
starts, the
minute.
infection essentially has ended. Now you might say that's interesting, but what happens if it's injecting DNA where we don't have a spacer that matches? Well, the CRISPR-Cas system works there as well. It's going to create a different class of protein, a class 1 Cas protein. And what that'll do is it takes the DNA in, it breaks it apart, but more importantly, it takes that DNA and copies it into the CRISPR system. So what is CRISPR? It is spacer, repeat, but the spacers are essentially history of old infections, so we won't be infected again. This is exactly the way your immune system works on a much larger level. You're making antibodies and then white blood cells that'll envelop that invader. But what scientists thought.
Okay, madam. Everyone, please see. I paused it and continued. In case if you don't have the spacer RNA, if you don't have it, a fresh infection comes, system will take another fresh enzyme and will try to mimic that sequence from the pathogen and put it in the DNA. Suppose, for example, you want to, you suppose, in general, madam, if you attend some functions, if the other madam has a different locket, what you do, you'll try to remember that locket and come back to your home and, you try to take money from your own wallet and you go and buy the similar locket, right? That means you also will have what you call protection against the gold crisis. The same way, what happened, this host cell will try to take the picture of a pathogen, that means bacteriophage, take the small one and put it back into the DNA sequencing. Next time, whenever function occurs, how you will wear this new locket, then your next pathogen enters into your body, we'll use this CRISPR, the last one, which is a short palindromic repeat of the last one, and we'll try to degrade the pathogen. You see how system is trying to evolve more spacers based upon the new infections, right? This is actually this technology we are using for genome editing. Genome editing means we can cut any gene sequences we wanted based upon this guide RNA, a short stretch RNA. Makes sense? Actually, I want to tell to my fellow colleagues that you may be sitting in different parts of the Andhra Pradesh. I may be sitting in Hyderabad. There's no difference between you and me because we both have technology accessible right away in our palm. The internet is there, technology is there, students are there, all about us learning the new things. I'm repeating same thing. Science lo kotta vadiki, sahityanlo pata vadiki pradhanyatam ekkuva. So we have to learn new things. That is why our life is so exciting. We never get bored of our job because every day is new thing for us. The same way evolution. Exactly how RNA interference, exactly how CRISPR-Cas will work. The same way your whole immune system will work. Memory. You do infection one time and we'll try to take some lesson from the infection. You know what? First time, even if you get the COVID infection, you're never going to get COVID infection again. Even though you get it, you get mild symptoms, but you never get second time full infection. That is the power of our immune system. Remembrance and act according to its memory. Hope you got this one, and I'll move to the next one. Oh my God, what happened? Okay, so this is the development of the non-specific resistance, that means immunity, a specific immunity system. So now I'll ask one quick thing. The first line what you learned was intact skin, mucous membrane, and the secretions and normal microbiota. This is what microbiota is everywhere in our body, from eyes to mouth and skin, large intestine, and urinary tract. Also, we have a protection against the pathogens. So this is the dryness of the skin, the temperature, low pH of the skin, sebaceous glands, bactericidal expression by the sebaceous glands, and squamation. Like your skin keep on removing. Actually, our skin will peel off when we are taking the shower. Even though you don't use soaps and all, the skin will go on. So that is one of the protection mechanism. Okay. So now I want to show you something called eyes, blinking of the eyelids, and tear containing lysozymes and wax containing antibiotic complexes. All are protect us, your ears. That's why we don't get infections. And mucous membrane is also protecting against us, because mucous cells are rapidly dividing the flush out of the body along with attached bacteria. Even when urination also, so many bacteria will flush it out of our urinary tract, both in men and women case. And digestive tract, low pH will definitely act on them. That is how this low pH in the gut. That is why most of the crocodiles, most of the flesh-eating animals, they survive because of pH in the gut. So again, this is urinary tract, just I told you. So now respiratory tract, this is also first line of defense mechanism, how you evolve. So we have alveolar macrophages are there. They provide the protection against it. And right from the nostrils, you have the hair in the nostrils. And same way you have the ciliated epithelium. It will never allow the bacteria to come into our gut or our lungs. Even though bacteria escapes, still we can protect against them. Okay, now, this is most important, microbial antagonism, normal flora and invaders. For example, imagine if in your house, you have two siblings, like for example. So your husband gets his వాళ్ళ అల్లుడినో కోడలో మీ ఇంటికి తీసుకొస్తారు. మీ వైపు నుంచి కూడా మీ సిస్టర్ బ్రదర్ నుంచి అల్లుడినో కోడలో తెచ్చుకున్న మీ ఇంట్లో పెట్టారు. కానీ ఫస్ట్ వచ్చిన వాడు చాలా ఫస్ట్ ఆఫ్ ఆల్ ఈజ్ అ గెస్ట్ ఫర్ యువర్ హోమ్, బికాస్ ఈజ్ నాట్ యువర్ ఓన్ సన్ ఆర్ యువర్ ఓన్ డాటర్, బట్ దే కేమ్ ఫ్రమ్ యువర్ రిలేటివ్స్. ఫస్ట్ వచ్చిన వాడు కొంచెం అడ్వాంటేజ్ ఉంటుంది. వాడు ఏం చేస్తాడంటే ఆఫ్టర్ సెకండ్ టైం కానీ ఫెస్టివల్ తర్వాత కానీ ఇంకొక సెకండ్ కజిన్ వస్తే ఈ డామినేట్ చేస్తాడు వాడిని. అరే కూర్చో, ముందు కూర్చో, అన్ని వాడు రెగ్యులేట్ చేస్తాడు. సేమ్ వే these pathogens come into our body. First time some pathogen comes, it slowly after million years it becomes normal and symbiosis with us. Now suddenly another bacteria want to come into our body and stay. So this guy is sitting there, he never let that guy come in. So that is why most of the times we can protection for us. It is called microbial antagonism. Already one bacteria sitting in our body, it is tuned with our body. The second one want to come into our body. The first guest never let the second guy to settle in our body. So beautiful, right? That is why you see yeast vaginitis. Actually, administration of the broad spectrum antibiotics may kill only certain members of the normal flora. For example, if we will have some antibiotics in female reproductive system, female urinary tract, there will be a yeast. That yeast will die because of these biotics. That is why you get super infection. That means though you want to take protection, but super infection. Second one, Clostridium difficile in colon, diarrhea and colitis. So what happens when you take antibiotics? this clostridium difficile, which is there, actually normal flora, it will die because of your antibiotics. So when this is not there, we get a diarrhea. That is why whenever you have antibiotics, whenever you have antibiotics you're taking, doctor will give you B complex. Why? Anybody can why when you take antibiotics, doctor will give the antibiotics. Why? Sorry, B complex. Why?
Sir,
Because
because
can
compensate the loss of
antibiotics kill the gut bacteria, sir. Antibiotics kill the gut bacteria.
sir, antibiotics kills the gut bacteria. Gut bacteria involved in the production of vitamin B12.
Very good, madam. Sir, you are very close, but sir. She said that because this friendly bacteria will provide B complex to our body. Since friendly bacteria died because of this antibiotics, as you said, to compensate that, the doctor will give the B complex vitamins. So everything is logic, sir, in science, sir. Okay, thank you so much, madam. And I hope another faculty will also talk. That would be good. So, sir, this is actually so much. This I'll share with you, sir. This data, because of interest of the time. Let us move further. Okay. So now you could see this bacteria evolution and gram positive bacteria and gram negative. What is the difference between gram positive, gram negative, madam? Anybody in the group? Gram negative. Difference between gram positive and gram negative.
Sir, gram positive bacteria has teichoic acids in its cell wall and more peptidoglycan. When compared to the gram negative bacteria, it consists of lipopolysaccharides in its cell wall, and it doesn't consist of teichoic acids.
Okay, very good. This is see the same thing you're talking about. Teichoic acid will be there, right? Teichoic acid will be gram negative or gram positive?
Gram positive, sir.
Teichoic acid. Gram positive will have more peptidoglycan, right? Teichoic acid will gram negative or gram positive, madam?
Gram positive, sir.
Really?
Gram positive.
Okay. So what is the acid nature, madam? What is the charge of the acid?
Charge negative, sir.
Negative. So peptidoglycan charge, madam?
Positive.
Oh, N-acetyl, madam. N-acetyl. N-acetyl, glucosamine, N-acetyl, muramic acid.
N-acetyl glucosamine.
Yeah. N-acetyl muramic acid. This is the main reason, madam. Muramic acid. Acid negatively charged will try to protect us. Negative charge is so crucial. Okay, madam. So important. So most of these acids will be helping us protection against the pathogens. Peptidoglycan, that's what muramic acid. So this is layer, and lysozyme will degrade between these two, right? This is how that so many layers are evolved. And what I would like to tell you here is that the bacterial cell wall is not a constant. It is also trying to when you divide, new layers will form. Now, question for you: penicillin is there. Penicillin. How the penicillin will be effective in how penicillin will be effective? Madam, penicillin injection. Penicillin.
Breaks the class limits.
Huh?
It breaks the class limits. Breaks the class limits.
Okay, I mean, no, connected to this sequence, connected to this one. Peptidoglycan. Connected to peptidoglycan.
Sir, penicillin can cleave the oligopeptides in between NAM and NAG, sir.
Excellent, sir. When it will cleave, sir? Sir, who said this question? Who said this answer, please? Who said this answer?
I'm Dr. Y. Polinaidu, GDC, Srikakulam.
Srikakulam. Naidu garu. Excellent answer, sir. My question is that, you know what is the penicillin is a bacteriostatic or bactericidal?
Bactericidal, sir. Yeah.
Madam, I asked Dr. Naidu, you are telling. Okay. Yes, how can you say it's bactericidal, madam?
It can kill bacteria, sir. By cleaving that outer cell wall.
What is the difference between static and cidal, sir?
Sir, static.
It can survive, it can die, sir, like that. It can survive, it is static.
Sir.
The bacteriostatic means just inhibit the growth of bacteria. Bactericidal means it completely kills the bacteria.
Exactly, madam. That is what whole point. When cell is dividing, then only penicillin can act. When cell is staying like this, penicillin cannot act. bacteria when trying to divide, penicillin can act. But bacteria when it want to stay like that and keep on doing pathogenesis, penicillin cannot do anything. That is important thing. See, clarity is most important. Bactericidal and bacteriostatic definition is okay. Penicillin is acting, penicillin mode of action are said okay. But the most important thing is that penicillin can only deal with dividing bacteria, and it can never let the bacteria cell divide, but it cannot touch the static.
So excuse me, sir.
Yes, please.
NRC Visakhapatnam, sir. Sir, excuse me, sir.
Yes, sir. Yes, sir.
What about, sir? Sir, penicillin does not directly to cleave the peptidoglycan.
It will inhibit
Penicillin to
enzyme.
inhibit the, sir.
Enzyme.
Penicillin to inhibit the serine proteases. Serine proteases need for the synthesis of peptidoglycan of the bacteria. That is the mode of action by.
Yes, sir. Yes, sir. Yes.
penicillin,
Very good.
sir.
I couldn't go deep. Okay, this is like very good faculty from Visakhapatnam. Okay, I have a request. I got a request from your colleagues. They asked to play the video. Do you want me to play the video of the RNA and CRISPR-Cas again? Naidu garu?
Yes, sir. Yes, sir.
Do you want me to play the video because sir said that it was
Yes, sir. Initial part of it is muted, sir. Okay, sir. You can play it.
Yeah, play. I will play immediately, sir.
Better
Sorry
idea.
for the inconvenience I caused it to you.
RNA interference is a mechanism of silencing gene expression after the gene has been transcribed. This interference is due to the presence of antisense RNA, which is complementary to a specific cellular mRNA. The complementarity between the antisense RNA and the cellular mRNA causes the two molecules to bind to each other. The antisense RNA can come from three sources. First, it can be introduced into cells by researchers. Second, certain viruses may produce antisense RNA as part of their life cycle. And third, the integration of genes into chromosomes next to endogenous gene promoters can sometimes cause the synthesis of the antisense strand of RNA. The current model of RNA interference involves two steps: creation of an RNA-induced silencing complex, RISC, and then the degradation of cellular mRNA by the RISC. Creation of the RISC occurs when double-stranded RNA is found in the cell. This double-stranded RNA is recognized by an endonuclease known as dicer that cuts it into pieces that are 21 to 23 nucleotides long. An antisense RNA fragment can then associate with proteins to form the RISC. The silencing complex can then associate with cellular messenger RNA that has a region of sequence homology with the RNA fragment in the RISC. The messenger RNA is then degraded. The presence of this rapid degradation mechanism can reduce and even effectively eliminate translation of the messenger RNA for the gene in question, thus interfering with gene expression.
Okay. See, Naidu garu, because I want to tell you in both cases what happens is that when a foreign mRNA comes in, our RNA interference mechanism try to form a small double strand RNA. Okay, in this case of RNA-seq, RNA interference. Same way CRISPR-Cas also, the bacteria trying to form a double strand RNA of the host one by using interspatial sequence of it and the long RNA of the pathogen. Like that, when they form the double strand RNA, even though it's small stretch, system will cleave it. The enzymes will cleave them. That is how, sir, system never let it to have a double strand RNA, right? That's what, no double strand RNA exists in the body, technically, right? That is how both will operate. Okay, sir. Now I'll try to take little of the movement further because okay, sir, these are the blood components. Closely look at the blood components, sir. Basophils, they have sir, one more important thing I want to tell you now. Few of these cells are technically, they are also represent our primitive microRNA because they have a natural killer cells. They can bind and they can imbibe and kill the, like amoeba. Like amoeba comes and attacks on its prey same way. These are some cells like macrophages, monocytes. I mean, macrophages derived from monocytes, and neutrophils, they attack the cells. Now you carefully look at these formed elements in the blood. Neutrophil and eosinophil. I want to show you something. If you want to identify these things, the trick is there. So for example, look at them now. Can you identify these leukocytes? What are the leukocytes, sir? Leukocytes are what are the leukocytes? Monocytes and lymphocytes are the leukocytes, right? You can see all of them. I just want to ask you, Naidu garu, meer cheppandi, top right side up undi kada, oka cell 3 lobes vachi, adi em cell anukuntunnaru meer? What you think, which cell is that? మీది మ్యూట్ లో ఉంది సార్, మీ యొక్క స్క్రీన్ మ్యూట్ లో ఉంది.
Sir, it is a multi-lobed nucleate, sir. I think it is neutrophil, sir.
న్యూట్రోఫిల్.
Yeah.
కింద చూడండి, పెద్ద సెల్ లో పెద్ద న్యూక్లియస్ ఉంది, చూడండి ఒకసారి. అదే స్క్రీన్ లో ఆ కార్నర్ లోనే, అందులోనే ఆ రైట్ కార్నర్ 2 ఉన్నాయి సార్.
Yes, sir. I think it's
actually let's see what you said.
monocytes, sir.
Okay, I'll tell you, sir. Actually, see, this is how you can make your student inquisitive, sir, because everybody know blood cells are చూసుకొని వస్తారు వాళ్ళ స్టూడెంట్స్ మీ క్లాస్ కి. చూసిన తర్వాత వాళ్ళకి ఇది న్యూట్రోఫిల్ అని చెప్పొద్దు, ఇది మోనోసైట్స్ మీరు చెప్పొద్దు సార్. ఫస్ట్ ఈ సెల్స్ ఎలా ఉంటాయని చూపించండి అంతే. జస్ట్ మీరు అడగండి క్వశ్చన్ డైరెక్ట్ గా వాళ్ళని. లెట్ us see how they will tell the answers. That's how you will help them to understand it, sir. So what happens, based upon that, you will tell them that some inquisitiveness. మీరు ఆన్సర్ చెప్పేసిన తర్వాత అడిగితే ఏంటంటే వాళ్ళలో ఆ ఇన్క్విజిటివ్నెస్ పోతుంది. మీరు కరెక్ట్ గా చెప్పారు సార్, వాళ్ళు మల్టీలో కరెక్ట్ గా చెప్పారు. ఎందుకంటే మిమ్మల్ని అడగకూడదు కదా క్వశ్చన్స్. ఇట్స్ నాట్ లైక్ మీరు ఆల్రెడీ ఫ్యాకల్టీ కాబట్టి ఐ డిడ్ నాట్ ఆస్క్ క్వశ్చన్, బట్ దిస్ ద ట్రిక్. This is always keeping the people interested in making the things beautiful. So now I'll try to show you something, sir. This is most important, sir. Evaluation of the lymphoid organs, sir. Immune organs ఎలా evaluate చూడండి సార్. జాలస్ వర్టిబ్రేట్స్ అంటే తెలుసు కదా సార్, అవి వాటికి జా ఉండదు, డైరెక్ట్ గా మింగేస్తాయి సార్ అవి. ఇప్పుడు చూడండి, ఇది ఇందులో గట్ అసోసియేటెడ్ లింఫాటిక్ టిష్యూ ఉంది. గట్ అంటే సార్ ఈ కడుపు లోపల ఒక లింఫాటిక్ టిష్యూ ఉంటుంది, అందులోనే మైక్రో ఆర్గాన్స్ అనిపేసే క్వాలిటీ ఉంటుంది అన్నమాట. లింఫాయిడ్ ఆర్గాన్స్ అంటే small. No, it's not a blood, sir. It is something different from the blood system. అదేం చేస్తుంది, ప్యారల్ గా ఉంటుంది బ్లడ్ కి, బట్ స్మాల్ సిస్టమ్, చిన్న బ్లడ్ వెల్స్ ఉంటాయి. అవి బ్లడ్ తో కంపేర్ చేస్తే చాలా చిన్నది అనమాట. లింఫాటిక్ టిష్యూ ఉంటుంది మనకి. ఇక్కడ ఉంటాయి కదా సార్, ఇవి టాన్సిల్స్ అంటారు చూడండి. టాన్సిల్స్, గజల్లో బిళ్ళలు ఉంటాయి, గొంతు, ఇవన్నీ లింఫాయిడ్ అనమాట. స్ప్లీన్ కూడా ఒక లింఫాయిడ్ ఆర్గాన్. ఇక్కడ స్ప్లీన్ ఉంటుంది కదా సార్ చెస్ట్ కింద లోపల, అవన్నీ లింఫాయిడ్. మీరు చూడండి కేర్ఫుల్ గా, ఇక్కడ చాలా కేర్ఫుల్ గా చూడండి. జాలస్ వర్టిబ్రేట్స్ లో గాల్ట్ అనే వచ్చింది, గట్ అసోసియేటెడ్ లింఫాటిక్ టిష్యూ. అదే ఫిషెస్ ఉన్నాయి కదా సార్, అవి కూడా తినేస్తాయి. కానీ ఫిషెస్ లో గాల్ట్ ఉంది, అలానే థైమస్, స్ప్లీన్ ఉంది. ఈ థైమస్ వల్ల ఎగ్జాంపుల్ ఏంటి సార్ అంటే టీ సెల్స్ వస్తాయి సార్, స్ప్లీన్ లో బి సెల్స్ వస్తాయి సార్, అలా వచ్చింది సార్. నెక్స్ట్ యాంఫిబియన్స్, ఈ మధ్యలో ఎవాల్వ్ అయినాయి కదా సార్. ఈ ఎవాల్వ్ అవుతున్నప్పుడు యాంఫిబియన్స్, ఇందులో గట్ అసోసియేటెడ్ ఉన్నాయి, థైమస్, స్ప్లీన్ ఉంది, బోన్ మ్యారో ఉంది సార్. మీకు ఒక ఎగ్జాంపుల్ చెప్తా సార్. ఇప్పుడు గట్ ఎందుకు ఉంటుందంటే, ఆ గట్ లో ఉన్న లింఫాటిక్ టిష్యూ డైరెక్ట్ గా మైక్రోబ్స్ చంపేస్తాయి మన తిన్న ఫుడ్ లో. ఎందుకంటే జాలస్ వర్టిబ్రేట్స్ అవి చెత్తలో ఉన్న ఫుడ్ కూడా అలా తినేస్తాయి, డర్టీగా ఉన్న వాటిని కూడా. ఫిషెస్ కూడా తింటాయి, కానీ ఫిషెస్ లో థైమస్ టీ సెల్స్ ఉన్నాయి. యాంఫిబియన్స్ ఉన్నాయి సార్. యాంఫిబియన్స్ అవి కొన్ని అటు ఉంటాయి, కొన్ని ఇటు ఉంటాయి. ఓకే, యాంఫిబియా అవి ఏం చేస్తాయి, వాటిల్లో బోన్ మ్యారో కూడా. బోన్ మ్యారో అంటే బి సెల్స్ వచ్చినట్టే కదా సార్, అంటే వాటిలో యాంటీబాడీస్ ప్రొడ్యూస్ అవుతాయి. అట్లానే రెప్టైల్స్ చూద్దాం సార్. రెప్టైల్స్ మీకు తెలిసిందో చాలా డర్టీయెస్ట్ క్రీచర్స్ సార్. అందులో చూడండి, రెప్టైల్స్ లో అన్ని ఉంటే లింఫ్ నోడ్స్ కూడా వచ్చింది. ఓకే, సో తర్వాత వచ్చేసి ఏవ్స్ అండ్ టెంపుల్స్ లో యూ హ్యావ్ లింఫ్ నోడ్స్ అండ్ జెర్మినల్ సెంటర్స్ సార్. అంటే మోర్ సఫిస్టికేట్ మనలో వచ్చినాయి సార్. ఎలా వస్తాయి సార్ ఎవాల్వ్ కాకుండా అంటే, మీరు లింక్ ఎలా ఉండనాయి, మన మెమల్స్ ముందు బర్డ్స్ ఉంటాయి, బర్డ్స్ ముందు రెప్టైల్స్ వచ్చినాయి, రెప్టైల్స్ ముందు యాంఫిబియన్స్ వచ్చినాయి, యాంఫిబియన్స్ కి రెప్టైల్స్ మధ్యలో ఆర్కియోప్టెరక్స్ అనే ఒక యానిమల్ వచ్చింది. ఆ యానిమల్ లో బోత్ రెప్టైల్స్ ఫీచర్స్ ఉన్నాయి, బర్డ్స్ ఫీచర్స్ ఉన్నాయి. బర్డ్స్ కి రెప్టైల్స్ మధ్య ఆర్కియోప్టెరక్స్ అని వచ్చింది. ఈ ఎవల్యూషన్ లో ఏమవుతుందంటే, ఫస్ట్ ప్రిమిటివ్ గా ఉన్న గట్ వచ్చింది సార్, బాగా గట్. ఆ గట్ లో ఉన్న వాటిలో లింఫాటిక్ టిష్యూ వచ్చింది. మీరు కావాలంటే చూడండి, లింఫాటిక్ టిష్యూ ఇలా ఉంటుంది సార్. ఇది ఈ లింఫాటిక్ టిష్యూలో ఏమవుతుందంటే, క్యాపిలరీస్ కి బ్లడ్ మధ్యలో వస్తుంది. ఇప్పుడు చూడండి సార్ కేర్ఫుల్ గా, లింఫాటిక్ టిష్యూ మీకు హార్ట్ కి బ్లడ్ తీసుకొచ్చేవాడు వీనస్ బ్లడ్, బ్లడ్ తీసుకెళ్లేవాడు ఆర్టరీ బ్లడ్. మీకు ఎప్పుడైతే హార్ట్ అనేది ఎవాల్వ్ అవుతుందో, ఈ లింఫాటిక్ టిష్యూ ఎవాల్వ్ అయిపోయింది. అంటే బ్లడ్ సర్క్యులేషన్ ఉన్న ప్రతి యానిమల్ లోనే వచ్చింది. చిన్నగా ఏమవుతుందంటే ఈ ప్రెజర్ వల్ల కొంత లిక్విడ్ బయటకు వచ్చి ఆ లిక్విడ్ డ్రైన్ అయిపోయి చిన్న ట్యూబ్స్ గా ఫామ్ అయ్యి మళ్ళీ అవి తెచ్చి వీనస్ సిస్టమ్ లో కలుస్తాయి. అంటే వీనస్ బ్లడ్ అయినా సరే ఆర్టరీ బ్లడ్ అయినా సరే ఆర్గాన్ దగ్గర కొంచెం ఊజ్ అవుట్ అయ్యి బయటకు వచ్చి లీక్ అయిపోయింది. మళ్ళీ చిన్న సిస్టమ్ ద్వారా లింఫ్ గ్లాండ్స్ ద్వారా కలెక్టింగ్ సిస్టమ్స్ ద్వారా లింఫ్ నోడ్స్ ద్వారా అది మళ్ళీ పూర్తిగా లింఫాటిక్ సిస్టమ్ ఫామ్ అయింది. ఆ లింఫాటిక్ గ్లాండ్స్ ఏనండి ఇవి మీకు వచ్చిన ఇన్ఫెక్షన్ వచ్చింది అనుకోండి వెంటనే ఈ లింఫ్ గ్లాండ్స్ స్వెల్ అవుతాయి. మీరు చిన్నప్పుడు ఎప్పుడు చూసారా గజ్జల్లో స్వెల్లింగ్ వస్తుంది, లేకపోతే ఇప్పుడు థ్రోట్ ఇన్ఫెక్షన్ వచ్చిన వాళ్ళకి టాన్సిల్స్ వచ్చినాయి అంటారు. ఈ టాన్సిల్స్ ఏం చేస్తారంటే లైట్ గా వేడి చేస్తారు. ఒక చిన్న ఒక వుడ్ని వేడి చేసి ఇక్కడ పెడితే ఆ యాక్టివేట్ అవుతారు అక్కడ. అంటే మోస్ట్ బ్యూటిఫుల్ ఫైటింగ్. ఇప్పుడు పాండవులు కౌరవులు ఇద్దరు కలిసి యుద్ధానికి ఎక్కడ చేద్దాం కురుక్షేత్ర ప్లేస్ లో యుద్ధం పెట్టుకున్నట్టు మన బాడీలో ఉన్న ఇమ్యూన్ సెల్స్ యాంటీబాడీస్ మన బాడీలోకి చొరబడ్డ ఈ ప్యాథోజన్స్ రెండు కూడా కురుక్షేత్రం అంటే ఈ లింఫాటిక్ గ్లాండ్స్ లో ఫైట్ చేస్తాయి. స్లోగా మనం వాటిని అటాక్ చేస్తాం. కానీ అటాక్ చేసినప్పుడు యుద్ధం జరిగినప్పుడు పెయిన్ వస్తుంది. ఆ పెయినే మనకు వచ్చే పెయిన్. మనం ఓర్చుకోకుండా ఏం చేస్తాము టాబ్లెట్లు వేసుకుంటాం. సో ఇఫ్ యూ వెయిట్ ఫర్ లిటిల్ లాంగర్ వీ గెట్ మోర్ ప్రొటెక్షన్, విల్ గెట్ ఎవాల్వ్ బెటర్. మీరు పెద్దానికి టాబ్లెట్స్ ఇస్తూ పోతారనుకోండి మన బాడీ ఎవాల్వ్ అవ్వదు, ఇమ్యూన్ సిస్టమ్ ఎవాల్వ్ చేసుకోలేదు అనమాట. సో దీస్ ఆర్ ద కాంప్లెక్స్ బట్ నేచురల్ ఎవల్యూషన్ హ్యావ్ టైనీ ఆర్గానిజం. నేను ఒక క్వశ్చన్ అడుగుతాను నాయుడు గారు మిమ్మల్ని, మనం స్మార్ట్ మనకన్నా చిన్నదాన్ని ఈక్వల్
పడండి సార్.
మనం స్మార్ట్ సిస్టమ్ ఎవల్యూషన్ లో మనం మెమల్స్ ఆర్ స్మార్ట్ ఆర్ ఈక్వల్ స్మార్ట్?
Equally smart, sir. I'm always with microorganisms only because even though they have single cell, they can perform all the functions and they can cause harm to us.
You're right, sir. Perfect, sir. Thank you, sir. I hope you got. I can share this information. I don't want to bore you people with telling more stuff. What I'm trying to say, like, no, sir, cell level. This is the most crux, sir. Actually, మీరు డివిజన్ లో అంటే మన దగ్గర పూర్వకాలంలో ఒకడే చేసే ఫంక్షన్, ఒకడే అన్ని ఫంక్షన్ చేసేవాడు. ఇప్పుడు ఏమైందంటే ఒక్కొక్కడు ఒక ఫంక్షన్ చేస్తున్నాడు. ఎవల్యూషన్ లోవర్ ఆర్గన్స్ అన్ని పనులు ఒకటే చేసుకుంటుంది ఈక్వల్. కానీ మనలో ఏమవుతుందంటే వీఆర్ వెరీ పూర్ సార్. అందువల్ల ఒక ట్రిక్ చెప్తాను సార్. ఇక్కడ ఇప్పుడు బ్లడ్ సెల్స్ లో WBC నెవర్ లెట్ మంకీస్ ఈట్ బనానాస్. ఎక్కువ న్యూట్రోఫిల్స్ ఉంటాయి, తర్వాత కొంచెం తక్కువ లింఫోసైట్స్ ఉంటాయి, వాటికన్నా తక్కువ మోనోసైట్స్ ఉంటాయి, వాటికన్నా తక్కువ ఇస్నోఫిల్స్ ఉంటాయి, చాలా తక్కువ పర్సెంటేజ్ బేసోఫిల్స్ ఉంటాయి. అందుకే NLMEB నెవర్ లెట్ మంకీ ఈట్ బనానాస్ అంటే కోడ్ అనమాట. మీరు పర్సెంటేజ్ చూసుకుంటే బేస్డ్ అపాన్ దట్ ఇస్ అ ట్రిక్ అనమాట అది. ఓకే సార్. సో దిస్ ఈస్ మోస్ట్ ఇంపార్టెంట్ అండి. నేను చెప్పేది ఏంటంటే ఎవల్యూషన్ లో ఐ కెన్ టాక్ ఫర్ అవర్స్ అబౌట్ దిస్ సిస్టమ్ ఎవల్యూషన్. RNA ఇంటర్ఫరెన్స్, తర్వాత యుబిక్విటనేషన్ వచ్చిందండి, ఆటోఫాజీ. సెల్ ఎప్పుడైతే తన కంట్రోల్ చేసుకోలేదు, ప్రొడక్షన్ చేసుకోలేదో సెల్ అండర్గోస్ ఆటోఫాజీ అనమాట. అది అలా ఒక సిస్టమ్ ప్రాసెస్ చేసుకుంటుంది సెల్ తనకు తానుగా. అంటే ప్యాథోజన్ స్ప్రెడ్ కానివ్వకుండా. కొంతమంది చేస్తారు కదండి, ఇప్పుడు వాళ్ళకి సెల్ చాలా బ్యూటిఫుల్ సెల్, తను ప్రొడక్ట్ చేయలేదంటే హోల్ బాడీని చంపడం కన్నా ఆ సెల్ ఒకటి చనిపోతుంది అనమాట. అట్లా చేసుకుంటుంది. ఓకే సార్, ఐ థింక్ ఇట్స్ టైమ్ ఫర్ యువర్ లంచ్. సో ప్రాబబ్లీ ఐ హోప్ యూ గైస్ got any questions for me. I can happy to take or write me questions. I'll happy to address you, sir.
Sir, one more question from my side, sir.
Chappan, sir.
Sir, after listening your CRISPR-Cas9, is there any like that mechanism in our body, sir? Because in secondary response, we do get immunity for different diseases in due course because of our age, na? Day by day, we may face some.
సార్ నాయుడు గారు, RNA ఇంటర్ఫరెన్స్ రాని మనకు రావు సార్. ఎందుకంటే మన ఎవల్యూషన్ లో చాలా ముందుకు వెళ్ళిపోయాం, మనకు లేవు. కానీ స్ప్లీను తర్వాత ఇవన్నీ మనలో ఉన్నాయి. లైక్ మెమరీ సెల్స్, కాంప్లిమెంట్ సిస్టము, ఆటోఫాజీ, నేచురల్ కిల్లర్ సెల్స్, డెండ్రైటిక్ సెల్స్, యాంటిజెన్ ప్రెజెంటేషన్. మంది వేరే స్టైల్ సార్. యాక్చువల్లీ మంది హైయెస్ట్ సఫిస్టికేటెడ్, బట్ చాలా సేపు పట్టింది. మనం బతకాలన్నా చనిపోవాలన్నా టైం తీసుకుంటాం. మంది వెరీ స్లో ప్రాసెస్.
సో ఈ బ్యాక్టీరియాలో ఉన్నటువంటి టెక్నాలజీని మనం జీన్ ఎడిటింగ్ కోసం యూజ్ చేయడానికి ఉపయోగపడుతుంది.
బాగా క్యాచ్ చేశారు సార్, వెరీ గుడ్. అంటే వాటి ప్రొటెక్షన్ మెకానిజం ని మనం ఇంజనీరింగ్ చేసుకోగలిగాం, అడాప్ట్ చేసుకోగలిగాం. మరి జావా, C++ మనోళ్ళు తయారు చేశారు. కానీ అది మార్కెట్ లో 2వ రోజే మైత్రివనంలో అమీర్పేటలో మాత్రం మనోళ్ళు ఎమ్మడి నేర్చేసుకుంటారు. మనోళ్ళు డీనో క్రియేట్ చేయలేరు. అది ప్రాబ్లం.
ఓకే సార్, థ్యాంక్ యూ.
థ్యాంక్ యూ
సార్. నమస్తే సార్.
నమస్తే అండి.
Sir, I'm Dr. G. Swati from PVK NRC. Had a small doubt.
మేడం సార్ మాట్లాడుతున్నారు మేడం. ఒకసారి పాపం సార్ మీరు లాగేసుకున్నారు, డామినేట్ చేశారు. సార్ స్టార్ట్ చేశాడు. విశాఖపట్నం
Sir,
సార్
sir,
అనుకుంటా.
thank you,
ఒకసారి
sir.
మేడం ప్లీజ్ ఆయన కంప్లీట్ చేయండి ప్లీజ్.
సార్ హోస్ట్ డిఫెన్స్ మెకానిజం ఇమ్యూనిటీలో చెప్పారు కదా సార్. ఇప్పుడు మనం చేస్తున్న ప్రొఫెషన్ కి హోస్ట్ డిఫెన్స్ మెకానిజం కి రిలేషన్ ఉందంట. ఫర్ ఎగ్జాంపుల్ ఒక మున్సిపల్ వర్కర్ ఉంటాడు, డైలీ ఆ గార్బేజ్ కలెక్ట్ చేస్తా ఉంటాడు. అతనులో హోస్ట్ డిఫెన్స్ మెకానిజం వర్సెస్ లెక్చరర్ హూ స్టేస్ ఇన్ ఏ క్లీన్ ఎన్విరాన్మెంట్ అండ్ డాక్టర్ అండ్ నర్సెస్ హూ స్టేస్ అమాంగ్ పేషెంట్స్ విత్ ఇన్ఫెక్షన్స్ and another person with a sportsman with an good fitness. అంటే ఇవి మనం డిఫరెంట్ ప్రొఫెషన్స్ అవి హోస్ట్ డిఫెన్స్ మెకానిజం ని ఎట్లా ఇన్ఫ్లుయెన్స్ చేస్తాయి కదా, దాని మీద ఒక
అవును సార్. యాక్చువల్లీ మీరు చాలా పరిశుద్ధంగా ఉండడం ట్రై చేయకండి సార్. ఇంట్లో పర్వాలేదు, అంటే మీరు ఇంప్రూవ్ అవుతూ వెళ్ళండి పర్వాలేదు, కానీ మీ బేబీ పుట్టంగానే వాడిని సిల్వర్ స్పూన్ లో మాత్రం పెంచకండి. ఏమవుతుంది, ఇమ్యూన్ సిస్టమ్ డెవలప్ కాదు. మీరు పిల్లవాడిని ఆడనివ్వండి సార్, గ్రౌండ్కి వెళ్ళండి, ఆడనివ్వండి, వాడి తోటి వాళ్ళతో ఆడనివ్వండి. స్కూల్ కి మనం ఏం చేస్తాం సార్, హైయెస్ట్ పాష్ స్కూల్ పంపిస్తాం, ఎయిర్ కండిషన్ క్లాస్ రూమ్స్ కి. ఏం అవసరం సార్ యాక్చువల్లీ అసలు. గాట్ ద పాయింట్. ఇమ్యూనిటీ అంటే కావాలని
సార్, అంటే ఇప్పుడు ఒకప్పుడు మనం చిన్నప్పుడు మట్టిలో ఆడుకున్నాం సార్, ఆడనిచ్చేవారు. ఇప్పుడు పేరెంట్స్ ఏమవుతున్నారంటే అసలు ఆడనివ్వట్లేదు. స్కూల్లో గానీ ఎక్కడ మట్టిలో లేక ఏంటి ఈ ఆటలన్నీ.
మీరేగా సార్ పేరెంట్, మీరేగా పేరెంట్.
అదే సార్, నో 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 కాదు సార్, నేను ఏం చెప్తున్నానంటే మట్టిలో ఆడమనేది నేను చెప్తున్నా. అంటే ఇటీవల ఒక డాక్టర్ గారి ఆవిడ ఏం చెప్తుందంటే మట్టిలో ఆడనివద్దు అంటుంది. నేనేదో మామూలు మేము కామన్ మినిమం బేసిక్ ఇమ్యూనాలజీ టీచ్ చేసే ఒక జువాలజిస్ట్ గా మట్టిలో ఆడనివ్వాలి నేను అంటాను సార్.
కరెక్ట్, మీరే కరెక్ట్ సార్. సార్ మీరు ఏదైతే చేస్తూ బతికారో, చేస్తూ ఎవాల్వ్ అయ్యారో అదే మీ
అంతే కదా సార్. అదే అంటున్నా, ఈ మనం చేసే ప్రొఫెషన్ అనేది తప్పనిసరిగా హోస్ట్ డిఫెన్స్ మీద ఇన్ఫ్లుయెన్స్ చేస్తుంది అంటాను. అంటే ఐ మీన్
చాలా సార్, చాలా. సార్ ఎవరో పీవీకే నుంచి ఎన్ఆర్సీ పీవీకే అని ప్లీజ్ సార్ రైట్ చేశారు సార్, హ్యాండ్ రైట్ చేశారు. మేడం చెప్పండి మేడం.
Sir, you had said about latency period in viruses. Latency period in virus. Latency period in virus, HIV virus is about 10 to 15 years back, sir. You said.
మేడం మీ వాయిస్ ఎందుకో సరిగా వినిపించలేదు సార్, చాలా బాగా వినిపించింది. మీరు లాటెన్సీ అంటున్నారు, నాకు ఆ లాటెన్సీ తర్వాత సెంటెన్స్ అర్థం కావట్లేదు. కొంచెం నెమ్మదిగా అడగరా ప్లీజ్. చిన్నగా.
How can it continue with latency period the cell dies? If the cell dies, what happens? How it can continue with latency period?
మేడం మీరు నాకు అర్థమైన క్వశ్చన్ ఏంటంటే, నాకు అనిపించింది అంటే 100% వినిపించలేదు. ఆ లాటెన్సీని ఎలా ఓవర్కమ్ చేస్తారంటే మేడం, లాటెన్సీలో ఏమైతే చెప్పనా మేడం, ఇప్పుడు సపోజ్ సడన్ గా మీరు ఏదైనా ఫుడ్ తిన్నప్పుడు, యాంటీబయాటిక్స్ తీసుకున్నప్పుడు ఆ లేటెన్సీలో ఉన్నది ఎవరైతే రెగ్యులేట్ చేస్తారో తగ్గిపోయింది అనుకోండి, అప్పుడు పైకి వస్తాయి. ఇప్పుడు మీరు ఎన్విరాన్మెంట్, సపోజ్ మీరు నార్మల్ గా మీ ఊళ్ళోనే ఉన్నారు మేడం, సడన్ గా మీరు అమెరికా వెళ్ళారు, అక్కడ ట్రిగర్ అవుతారు అనమాట. ట్రిగరింగ్ ఫ్యాక్టర్స్ ఎప్పుడు ఉంటాయి. సపోజ్ మీరు ఎప్పుడు వంకాయ కూర తినరు, సడన్ గా ఇప్పుడు వంకాయ కూర తినగా సడన్ గా ఏమైందంటే వంకాయలో ఉన్న ఎలర్జీ అనేది మీ బాడీని ఎఫెక్ట్ చేసింది. అలా ట్రిగరింగ్ ఫ్యాక్టర్స్ ఉంటాయి. ఆ ట్రిగరింగ్ ఫ్యాక్టర్స్ వల్ల ఏమవుతుందంటే ఈ ఓవర్కమ్ చేస్తారు అనమాట. అలా లేటెన్సీలో ఉన్నది మళ్ళీ మెయిన్ స్ట్రీమ్ లోకి వస్తుంది. అట్లా జరుగుద్ది మేడం. మీరు పొద్దున్నే తుమ్ముతారు చూడండి, చాలా మంది పొద్దున్నే తుమ్ముతుంటారు. ఎందుకని? ఎలర్జీ. మామూలు సీజన్ లో ఉండదు, సమ్మర్ పోయి వింటర్ వచ్చే టైంలో ఆ తుమ్ము వస్తారు మేడం. డిఫరెంట్ ఎన్విరాన్మెంట్ క్యూస్ ఉంటాయి మేడం, అవి అందువల్ల లేటెన్సీ ఓవర్కమ్ అవుతుంది అనమాట. మేడం మీకు నా క్వశ్చన్ సరిగా అర్థం కాకపోతే మీరు కాల్ చేయండి మేడం, నేను డెఫినెట్ గా మీకు రెస్పాండ్ అవుతాను. పీవీఎన్ జీసీ, ఎన్ఆర్సీ పీవీకేఎన్ జీసీ మేడం. చెప్పండి సార్, చెప్పండి.
Regarding the appendix one, appendix which is having a secondary lymphoid, secondary lymphoid organ.
అవును సార్, అవును సార్.
Still regarding evolution, considering it as a non-functional organ by 6 law.
సార్ మీ క్వశ్చన్ సెకండరీ లింఫాయిడ్ ఆర్గాన్స్ వరకు అర్థమైంది సార్. సెకండరీ మీరు అడుగుతున్న ప్రిసైజ్ గా సెకండరీ లింఫాయిడ్ ఆర్గాన్స్ ఏమవుతుంది సార్?
Sir, appendix, it is having lymphoid tissue.
Lymphoid tissue, yes, sir.
So it is a function or the other. But still it is considered as non-functional organ regarding evolution. Reduced and non-functional.
Okay. No, sir. Sir, వాటి సైజ్ తగ్గిపోయింది, ఇంపార్టెన్స్ తగ్గిపోయింది, కానీ దే ఆర్ ఫంక్షనల్ సార్. పర్పస్ మనలో కూడా పేర్ ప్యాచెస్ అని ఉంటాయి సార్. లింఫాయిడ్ టిష్యూ మాల్ట్ మనలో కూడా ఉంది సార్. క్యూటేనియస్ లింఫాయిడ్ టిష్యూ ఉంది మనలో కూడా. అందుకే సార్ వినండి సార్, మీరు ఒకసారి కూడా ఇప్పుడు అడిగారు. ఇప్పుడు మనం కూడా హ్యాపీ సర్వే అవుతాం డర్టీ వెదర్ లో. కాకపోతే ఏంటంటే కొన్ని ఇమ్యూన్ మనకి లంగ్స్ లో కానీ కొన్ని ఇమ్యూన్ యాంటీజెన్స్ లేవు. అందుకే మనకి ఫస్ట్ ఇన్ఫెక్షన్ వచ్చింది కానీ ప్రొటెక్ట్ అవుతాం కొన్నాళ్ళకి. మనలో ఫంక్షన్ ఉంది సార్. కాకపోతే ఇంపార్టెన్స్ తగ్గుతూ వచ్చింది సార్. అంటే మనము సార్ ఇప్పుడు మనకి అపెండిక్స్ ఉంది కదా సార్, అపెండిక్స్ ఆర్గాను అది కూడా తగ్గుతూ వచ్చింది కదా సార్. అట్లా మనలో ఇంపార్టెన్స్ తగ్గిపోవడం వల్ల అలా జరిగింది. స్పెషలైజ్డ్ టేక్ ఓవర్ సార్.
Still can be called as non-functional organ and must be, sir.
Yes sir, yes sir, right, yes sir, you are right.
But it is having lymphoid tissue and being some function.
అంటే ఏం సార్, అంటే లింఫాయిడ్ టిష్యూస్ లో కూడా జర్మినల్ సెంటర్స్ లేవు సార్. వాటికి మనకున్న జర్మినల్ సెంటర్స్. అంటే లింఫాయిడ్ ఆర్గన్ లో కూడా కొన్ని స్పెషలైజ్డ్ ఇంకా ఫర్దర్ ఫర్ ఎగ్జాంపుల్ సార్, మీకు ఒక స్కూటర్ ఉంది సార్. నా స్కూటర్ లో హై సిసి ఇచ్చారు, ఆ తర్వాత హైడ్రోజన్ గ్యాస్ తో నడిచే స్కూటర్ ఇచ్చారు. అట్లా అంటే పాత వాటికి ఏమో ఓన్లీ స్కూటర్ పెట్రోల్ తో నడిచింది. మనకి ఇప్పుడు ఈ సొఫిస్టికేటెడ్ ఈజీగా నడిచేది. ఇంప్రూవ్ అవుతుంది, ఆర్గాన్ లో ఇంప్రూవ్ అవుతుంది, ఆ యొక్క పర్టికులర్ సెల్స్ ఇంప్రూవ్ అవుతున్నాయి. సో వీఆర్ మోర్ సొఫిస్టికేటెడ్, బట్ క్రూడ్ ఫంక్షన్ మాత్రం తగ్గుతూ వస్తుంది.
ఓకే సార్.
బాయ్. అంటే నాకు మీ కొంచెం వాయిస్ కూడా, నా వాయిస్ ఎలా అనిపిస్తుంది సార్? సార్ మై ఫోన్ నెంబర్ ఇస్ దేర్ సార్, మై ఈమెయిల్ ఇస్ దేర్. ఎనీ క్వశ్చన్స్ ప్లీజ్ ఆస్క్ మీ సార్, ఐఎమ్ హ్యాపీ టు అడ్రస్ సార్.
Yes. Thank you sir. Thank you so much.
Bye sir. Bye Andy. Bye everybody sir. Have a nice day sir.
Sir, this is Dr. Thank you very much, sir. Today you have given a very excellent lecture on the post defense mechanism, sir, from the point of evolution. Sir, sir.
Yes sir.
We are proposing one of thanks, sir, on behalf of all the NRCs, all the faculty who are participating in this session, sir. So it is a very good and elaborative and excellent lecture, sir. It is informative, ranging from the simple concepts like infections, colonization, and you have linked the cytokine storm to COVID deaths and all these things, sir. So finally, the lines of defense, first line defense, second line defense, and cellular defense systems. And that is the more advanced mechanisms derived from the body itself, from the point of evolution. And more important, you created emphasis in the participants using very good pedagogical techniques like interactive sessions and examples, sir. For all these things, on behalf of all the NRCs, we thank you very much, sir. Thank you, sir.
థ్యాంక్ యూ అండి, థ్యాంక్ యూ సో మచ్. ఉంటాను సార్, మళ్ళీ కలుస్తాను ఎప్పుడు ఫ్యూచర్ లో. ఇన్ ప్రెజెన్స్ అయితే బాగుంటది కానీ, బట్ సమ్ హౌ నో. ఓకే, థ్యాంక్ యూ అండి. హ్యావ్ ఎ నైస్ లంచ్. బాయ్ అండి ఎవ్రీబడి.
బాయ్ సార్.
బాయ్ సార్, థ్యాంక్ యూ.
Okay, sir. Lunch. Next session.
So what to take a speech?
Online technique maname attend cheyali.
Can we break for lunch?
So we will
be back by.
You can leave it.
Requested another Okay, I got. Sir, good afternoon, sir.
Hello, good afternoon, Srinivas. Can you hear me?
It is audible, sir. It is very much audible.
Okay, I will try to share the slides. Let me check.
Just a minute, sir. Some of the participants from other NRCs.
Okay, no problem. But last time what happened is that when I was trying to share, somehow I couldn't able to do it. I thought maybe we'll test once, then we.
The NRCs people will give you the access, sir, and only you can share, sir.
Okay, okay.
I'll get back to you, sir. In 5
Sure,
minutes.
sure, no problem, no problem. Okay. Hello, can you hear me?
Sir, it is audible, sir.
Yes, sir.
I request all the NRCs.
All of you please come.
Get ready. The resource person has already joined. Once again, I request all the NRCs. The KRPs, please see that the all participants will be there. The conference hall within 5 minutes. Research person has already joined and we will start our session.
వీటిలో పెట్టి వాడతారు
Yes, sir, my concession
I just wanted to check, can you hear me?
Madam, please, madam.
Yes, sir. Yes, sir.
So, because I hope this is clear, right? Okay.
Yes, sir.
Please respond, sir. Please introduce the research person to the audience.
Yes, sir. Srinivasan,
Yeah, please, please go ahead.
myself, Dr. K. Ramakrishnan, lecturer in Zoology, and I am representing SCM JDC Laboratory NRC. Srinivasan, welcome you to online session at Degree College NRC level across every state, and I also welcome all the KLC persons and participants, JDC coordinators. It is my privilege to introduce the resource person of life, Srinivasan. So, he is working as a professor in the Department of Animal Biology. Coming to his career, Sir completed MSc Biochemistry from SKUCT. Then later completed PhD in the Bananas University. Later he moved for the postdoc to the Sweden. And also he completed second postdoctoral research scientist in the Biotechnology Institute, Cambridge, UK. And also he continued as a research associate in the Institute of Cancer Science, University of Glasgow in the UK from 2008 to 11. Then next, he came back to the India and he joined as a reader for the Department of Animal Sciences, University of Hyderabad, India, and he served as a reader from 2011 to 13, and he later promoted as associate professor from 2013 to 18, and currently he is working as a professor from 2018 to till date. Then coming to publication, Sir has various publications in repeated journals like BMC Genomics, Cell Reports, BMC Research Notes, Veterinary Immunology, Nucleic Acid Research, Nature Protocol, Blood, etc. Then Professor Srinivasan is working in various projects. Few of the ongoing projects like efficiency regulation of gene expression by chromatin supercoid, implications in neuronal development and disease, and also one more project is ongoing is computational investigation into the role of alternative splicing in cellular differentiation, tissue regeneration, and evolution. There is a area of research in epigenetic mechanism of gene regulation during cell differentiation and disease. So, I am very thankful to give this opportunity to CCET and all the KRPs and all the participants. Thank you very much.
Okay, thank you, Dr. Srinivas. So, can I share my slides?
Yes, sir.
Okay.
Yes, sir, it is visible, sir.
Okay.
Slides are being sharing, sir.
Okay.
Eukaryotic genome organization structure and function.
Yeah, okay. Thank you very much. So, first of all, thank you very much for the invitation. It's my pleasure to make a presentation about eukaryotic genome organization, structure and function. As I understand, you all are zoology teachers, so I think it is very important for me because I also done a PhD in zoology. But you know, zoology is starting from classical zoological aspects to now I'm trying to look at much modern perspective. So with this brief introduction, I would like to proceed. So all of us know that. Hope you can see my cursor. See, life of an every organism from a single cell zygote that is formed by the act of fertilization by haploid oocyte and then haploid sperm, resulting in the formation of a diploid zygote, and then diploid zygote, which is a single cell, by means of a proliferation of cells and then differentiation, eventually comes to a morula stage and then blastula stage. And then within this blastula, as you can see at the interior of this particular blastula, there are cells which are called inner cell mass cells. These are stem cells. These are also called embryonic stem cells. These embryonic stem cells subsequently can give rise to each and every cell of the embryo, constituting various organs and organ systems. And eventually this embryo, depending upon the sex of the embryo, it can become a male organism or female organism. Again, they produce the germ cells of eggs or sperms, thus completing the life cycle. One of the fundamental questions in zoology, for that matter, one of the fundamental questions in development biology is that how come a single cell zygote, which is derived from the fertilization of a haploid oocyte and then sperm, can eventually give rise to such a complex organism? What is actually inside the zygote that eventually can make an individual organism? As we know that the zygote is basically formed by the fertilization act of a oocyte and sperm. But in some of the experimental conditions, if you try to make a zygote from a oocyte and then the other haploid nucleus from other oocyte, thus if you make a diploid oocyte, so it will not grow into a functional embryo. At the same time, if you take a oocyte and then if you remove the nucleus and then you introduce a haploid nuclei from two independent sperms into the enucleated oocytes, even that will not develop into an embryo. That means component of a male genome, that is oocyte, component of a male genome, that is in form of sperm, and then component of a female genome, that is oocyte, are both nuclei required for establishing a functional zygote that only subsequently develop and differentiate into an adult organism. This has given the indication that something else other than genomes from oocyte or sperm is coming that will play an important role for the functional development of a zygote. And now we know that these more than DNA, that is beyond DNA, that is very much important, which is there in the oocyte genome and then sperm genome, that is important for establishing a functional zygote. So in that context, that particular phenomena, which is beyond the genome per se, important for establishing the functional embryo, is actually derived from a phenomenon called epigenetic phenomena. So epigenetic phenomena here at the molecular level, the DNA from oocyte and then sperm, although they are having identical sequence, but they have some kind of DNA modifications that is called the methylation, which is unique for oocyte and unique for sperm. Hence, this kind of differential methylation of DNA of a oocyte and then sperm are very, very important for the formation of a functional zygote. So from here, I wish to let you know that. So we all know that from the single cell, we can give rise to such a complex organism. So if you just look into this early blastocyst stage, the cells that are present at the interior of this particular blastocyst is called inner cell mass. These cells has the capacity to give rise to each and every cell type of our body. Hence, they are called stem cells. And these stem cells, as seen here, to start with, they have the capacity to give rise to each and every cell type of our body. All cells of our body are basically derived from three progenitor cells, that is ectodermal progenitor cell, mesodermal progenitor cell, and then endodermal progenitor cells. Then all these three cell types are derived from embryonic stem cells. So embryonic stem cells, they are pluripotent because they can give rise to all these three germ layers, and then individual germ layers for ectodermal progenitor cell, then eventually give rise to neuron, glial cell, and epithelial cells. That means starting from pluripotent stem cells, you will have a multipotent progenitor cells. From multipotent progenitor cell, you will have a differentiated cells. And then one of the very fundamental questions, and we know that all the cells of a human body derive from these three germ layers, ectoderm, mesoderm, and endoderm. And all these ectoderm, mesoderm, and endodermal progenitor cells basically derived from inner cell mass stage of early blastocyst stage. And then one of the fundamental questions that although all cells of our body are derived from embryonic stem cell, all of them are having identical genomes. And then in spite of having identical genomes, how that each and every cell is very unique. And we know that liver cell transcribes liver specific genes and they have liver specific proteins. Neurons, for example, they transcribe neuron specific genes and then neuron specific proteins. Thus it manifests as its function. But fact is that all these differentiated cells are having identical genome. So instead of having identical genome, how that a particular cell knows that it is so and so cell, and they have to transcribe and then translate a few subset of genes. For example, in humans, our genome codes for nearly 30,000 genes. Out of these 30,000 genes, each and every cell transcribes only a few subset of genes. For example, liver transcribes around 6,000 genes, and then neuron transcribes around 5,000 genes. But if you see the genes that are transcribed in neuronal cell and then liver cell, they are very different. So that means, although all cells are having identical DNA sequence per se, but each and every cell transcribes and translates different sets of genes, and these different sets of proteins that are there in each and every cell, that is actually manifest as different functions to different cell types. Neurons, skin cell, liver cell, kidney cell, cardiomyocytes, they all are unique because in terms of their gene expression patterns, but in spite of the fact that all of them are having identical genome. So one of the fundamental questions is that how come in spite of having identical genomes in all the cell types, how come that every cell transcribes a different sets of genes and then proteins? Okay, so before going into that kind of fundamental aspects of regulation of gene expression, let us see, we know that in eukaryotes, the genetic material deoxyribonucleic acid in the form of a chromatin is present within the spherical nucleus. And we know that, for example, in case of human cells, our genome has can be seen in the form of a chromosome, and humans are having 23 pairs of chromosomes. So 23 chromosomes coming from mother, and then 23 chromosomes coming from father, and then all these 46 chromosomes are present within the spherical nucleus. That means each and every chromosome has a single DNA molecule. So if you try to put together all these DNA molecules, we have approximately 2 meters length of DNA present in 10 micrometer volume of a nucleus. I hope you all are hearing my voice. Okay. Yeah. So our genome is present within the nucleus, and then if you see here is a cell where the nucleus is spherical, and you can see the blue color. Here we are seeing them as nucleus as a blue color because there is a dye called DAPI which is stained with this particular cells. DNA takes up this particular DAPI. Then when you observe under fluorescent microscope, you can see them as a fluorescing in blue color. So you can see that in one of the cell, the DNA is highly diffused toward the nucleus. In other cell, which is actually undergoing cell division, you see that chromosome-like organization. That means our genome within the nucleus can sometimes appear like a chromosome, sometimes it is present in the diffused pattern. And then within this particular nucleus, so if you try to take out the chromatin and then try to observe under electron microscope, it can be looked in the form of a beads on string-like form, or sometimes it can also form a much higher order folded, much very dense chromatin fiber. So here is the picture showing you the structure of the DNA, and then at the very primary level, we know it is made up of ATGC, and then DNA in association with the histone group of proteins forms a nucleosomes. So nucleosomal DNA is called chromatin, which is actually called the nucleosomal chromatin. And then in higher order structures, this chromatin can be folded in higher order folder and then thus appears like a chromosome at particular stage of cell cycle. So this picture actually show you if you try to take sections of the cell nucleus where the genetic material is present, you can see these kind of structures where here you can see the cross section of a typical mammalian cell nucleus where you see the electron dense heterochromatin which is seen towards the nuclear periphery, where the electron poor euchromatin is seen towards the interior. That means we have a compartmentalization of chromatin within the cell nucleus. The dense heterochromatin, which is rich for silent genes, they are present in the nuclear periphery, whereas the decondensed or electron poor decondensed euchromatin, which is rich in genes which are transcriptionally active. So at the same time, this is the cell nucleus during interphase. Same cell, when you try to observe during metaphase stage, you see that they appear like a beautiful X-shaped chromosomes. So we know that each X-shaped chromosome is nothing but it is the DNA and then chromatin is condenses, appears like a rod-shaped chromosome. This is basically duplicated chromosomes. So chromosomes, X-shaped chromosomes are nothing but these are duplicated chromosomes. This is what exactly happens during process of mitosis. So if you try to see within this X-shaped chromosome, how actually the DNA molecules are present. So next slide will show you how exactly DNA is actually placed within this X chromosome. So we all know that the DNA structure, right-handed double helical structure, during replication, we know this original strand. Thus, this orange colored DNA representing the newly synthesized strand. So thus, starting from a single DNA molecule, at the end of the replication, we have two DNA molecules. Each DNA molecule retains one of the original strands of DNA, and then each DNA molecule now has one newly synthesized DNA. This kind of DNA structure, if you try to see in this X-shaped chromosome, in this X-shaped chromosome, as you can see, the red color shows the older DNA strand. Each chromatin retains the one older strand, and then newly synthesized green strand is seen along with the arm. So this kind of understanding is very, very important for us to understand the mechanisms of entire genetics and then its segregation during mitosis or meiosis process. So within this particular nucleus, we have a compartmentalization of genes, heterochromatin, euchromatin. If you look into individual genes, so by definition, every gene, as shown here, you have every gene has a promoter, and then every gene starts with exon, and then followed by intron. Because in eukaryotes, genes are called split genes. We have an exonic as well as intronic portion. So when this particular DNA undergoes transcription by proteins called RNA polymerase II, which converts DNA to RNA, you see that RNA is being synthesized, representing both exonic as well as intronic portion. Exons are here depicted as a blue color, and the introns are depicted as red color. And then within the nucleus, after transcription, this primary transcript undergoes a phenomenon called splicing. Thus, selectively, all the exonic component within the RNA are eliminated, and then introns are eliminated, and then all exons are fused together by matured messenger RNA, which comes out of the nucleus within the cytoplasm, which undergoes a phenomenon called translation. Thus, RNA is codes for a particular protein, and this protein assembles in a three-dimensional structure. That's where the proteins get some kind of function. Depending upon the kind of proteins that a particular cell has, then that is exactly dictates the functions of a cell. And then within this gene context, so this is typical gene of a particular eukaryote. These genes are actually regulated by certain regulatory elements such as enhancer. So for example, here in this particular middle figure, as you can see, you have a gene with promoter P1, and there is a regulatory element called enhancers. Enhancers are the sequences which are present away from the gene body, but whose presence is important for transcriptional activation. So when the gene, yes, please. So can you hear me?
Yes, sir.
Oh, okay, sorry. Yeah. As you can see in the middle figure, when the gene is transcriptionally active, it distally placed enhancer sequences where certain proteins are bound, they loop away, Thus, the distal enhancers are seen near to the gene promoters. Thus, when they come together, then gene will undergo transcription. So in absence of this enhancer promoter communication, no eukaryotic gene is transcriptionally active. And then further, for example, in this other third figure, as you can see that you have a chromatin fiber where you have a genes and then regulatory elements. So one of the gene, which is depicted in the blue color, as you can see that in the nucleus, it forms this kind of loop organization called topologically associated domains. So this kind of organization will restrict a particular gene promoter and gene enhancer to be communicated specifically. So to just tell you that in eukaryotes, in order to genes undergo transcription, they require regulatory elements such as enhancer, which is actually present away from the gene body, but during transcription activation, enhancer can be seen to the near to the promoter, thus genes can undergo transcription process. Okay, so research from the several years, now it has shown that, so although all cells are having identical DNA, but we know that each and every cell transcribes a cell type specific genes. Now, with the help of multiple genomic studies, now scientists have discovered that there exists some kind of code or some kind of signatures for active chromatin and then silent chromatin. So we know that each and every cell type has a unique silent genes and then unique active genes. If you look into those genes which are transcriptionally active, it is right side on. So those genes which are transcriptionally active in a particular cell type, their DNA is devoid of a epigenetic mark, that is DNA methylation. So all those genes which are transcriptionally active in a particular cell type, they are devoid of DNA methylation, which is actually the code for transcriptional activity. At the same time, if the DNA is heavily methylated, that is most likely that that gene is repressed in a given cell type. Similarly, if the gene is off in a particular cell type, you have a regularly placed nucleosomes over the gene, and then when the gene is transcriptionally active, there you will find the irregularly placed nucleosomes over the gene body. And at the same time, like a DNA undergoes methylation, even those proteins which are made up of these nucleosomes, histone proteins, they also undergoes some kind of post-translational modification such as methylation. And methylation of particular histone proteins are very uniquely placed for active genes and then silent genes. For example, all those genes which are transcriptionally active in a given cell type, they have this important histone modification such as H3, K4 trimethylation, histone H3, lysine 4 position undergoes trimethylation. At the same time, histone H3, K36 trimethylation. These are hallmarks for all the genes which are transcriptionally active in a given cell type. At the same time, all those genes which are repressed in a given cell type, they have other special modifications such as histone H3 K9 trimethylation and a histone H3 K27 trimethylation. So that means there exists some kind of code within the chromatin that will ensure that genes are either transcriptionally on or transcriptionally off. At much higher order level, you can see that all the silent chromatin is highly condensed and then seen at the nuclear periphery, whereas the highly active chromatin is decondensed and then which is seen at the nuclear interior. So these are the hallmarks for the genes that will tell you whether a particular gene to be transcribed or not in a cell type specific manner. For example, insulin gene is present in each and every cell of our body physically, but insulin gene transcribes and then translates only in islets of Langerhans of pancreatic cells. So if you try to look at the features of the insulin gene in islets of Langerhans, its gene body is devoid of DNA methylation, its nucleosomes are irregularly placed, and they have the special modifications H3K4 trimethylation, H3K36 trimethylation, and then their chromatin can be bound with RNA polymerase II. Whereas same insulin gene in all other cell type, they have a signatures for transcription off state, such as their DNA is heavily methylated, nucleosomes are regularly placed, and then they have this special silencer modification, and then their chromatin is highly condensed and then pushed towards the nuclear periphery. That means someone is already created some kind of epigenetic information within the cell in the form of histone modifications or DNA methylation or nucleosome positioning or higher order chromatin that will ensure that each and every cell knows what kind of genes to be transcribed in a cell type specific manner. Okay, further just to go deep into this particular field, see this is what exactly typical nucleus looks. You see that heterochromatin, which is seen in the darkly electron dense material, seen in the nuclear periphery, and within nuclear interior you can find the euchromatin. And we know that humans were having 46 chromosomes. Each and every chromosome has one DNA molecules. Okay, so then that every nucleus of a human cell, we must have 46 DNA molecule. So in this particular picture that what I'm showing you here, if I ask you a question where the DNA belongs to a particular chromosome is present within the this particular space, and then when the scientists asked this question and then they performed a technique called DNA fluorescence in situ hybridization by using the probes for a chromosome 1 and chromosome 2 DNA, as you can see here is that as you can see here, for example, chromosome 1 DNA is labeled with red color, say chromosome 2 DNA is labeled with green color, and then they use DNA fish technology to detect the DNA belongs to a particular chromosome within the interface cell nucleus. Surprisingly, they did not find any chromosomes, but surprisingly, chromosomal DNA belongs to a chromosome is not randomly distributed throughout the nucleus. You can see that DNA belongs to each and every chromosome occupies a unique territory within the nucleus. These are called chromosome territories. Okay, so that means if chromosome 1 here the blue color, you can see the two locations where there is a blue color is coming up. That means since we are diploid, one chromosome territory must be coming from a mother, other chromosome territory must be coming from father genome. Similarly, other chromosome which is labeled with red color, as you can see that they also seen at the two different locations, and one must be from mother chromosome, other must be from father chromosome. So within the nucleus, the DNA belongs to a particular chromosome interface is not appearing like a chromosome, but their DNA is highly diffuse, but it's not diffused throughout the nucleus, but it is diffused and occupies a constrained location within the three-dimensional space of the nucleus are called chromosome territories. Okay, so within these chromosome territories, for example, here I'm showing the single chromosome territory. As you can see that there is a blue region and then green region, but it is basically linear polymer. The green region is the heterochromatin, blue region is the euchromatin. As you can see that all the heterochromatin, where you have a silent genes, they are clustered and then pushed toward the nuclear periphery, whereas all the active genes within a chromosome territory, which are placed towards the nuclear interior, and they are highly decondensed. And then by using this kind of DNA fish technology, scientists learned that in interface cell nucleus, DNA belongs to every chromosome, occupies a unique territory called chromosome territories. But interestingly, when they try to detect multiple chromosomes at a time by labeling different chromosomes with different fluorophores, they learned that none of the chromosomes are seen together, homologous chromosomes. They always have some specific location. From these studies, they observed a phenomenon called, if you try to do this kind of technique in one particular cell type, they see that the positioning of these chromosome territories are similarly placed in all cell types of a particular cell type. If, for example, if in the liver, you have this kind of pattern. If I see the same kind of technique in neuronal cells, they learn that this pattern is different. If I try to do this in a skin cell, again, the pattern is different. From these studies, they learned that chromosome territory positioning is specific to each and every cell type. Then further, by using a technique called spectral karyotyping. Spectral karyotyping is the method by which one can locate each and every chromosome is differently labeled with some kind of fluorophore. So you can identify each and every chromosome based on its color. So similarly, during interface, chromosomes are not exist as chromosome territories, they exist as a chromosome territories. So you can identify the chromosome territories in a cell type specific manner. So by using this kind of spectral karyotyping, scientists have analyzed chromosome territory positioning of various cell types of human body. From these studies, they learned that each and every chromosome is uniquely placed in a cell type specific manner. That means chromosomes are radially positioned and their organization is cell type specific and is linked with the differential gene expression fragment. So here the beauty here is that although all cells are having identical genome, but the way each and every chromosomal DNA is placed and then it has a neighboring chromosome territories is unique for each and every cell type. Okay, so you can see that it is very puzzling. It was really, when scientists realized that this was the case, they are really puzzled. So now that makes a sense why that liver cell is a liver, why neuron cell is a neuron cell. All the brain cell and then liver cell have identical genome, but the way the chromosome territory positioning and then neighborhood are placed among these cell types is very unique. They're unique for each and every cell type. Okay, but how scientists came across this kind of phenomena? How did they learn that chromosomes are having cell type specific chromosome territory organization, and then within the chromosome how the DNA is organized, and then how that actually genes are seen within the chromosome territories? So this all important information comes from a technique called a molecular tool to understand the physical organization of genes, chromosomes, and the genome. So these techniques are named here. For example, chromosome conformation capture. And the next set of experiments methods are chromosome conformation capture on chip. In short, they are called 3C. And then there is one more high throughput method called Hi-C. All these methods are very, very important, which were developed from the past over the period of 10 years. These techniques are very important, that these techniques actually told us how the genes are actually placed within the nucleus when they're transitionally on, how they're actually physically organized, how they're transitionally off, how they're actually organized, how that each and every gene is present within the chromosome territory, how that various chromosome territories you can find active euchromatin, active heterochromatin. All such kind of information actually gathered, scientists have gathered by using these powerful technologies, 3C, 4C, and Hi-C. As I showed here, 3C is the technique which is very much helpful to understand the enhancer promoter communications. So you can identify a particular gene and its putative regulatory elements such as enhancer. And by using 4C, you can also can understand what kind of genes are present within the vicinity of a given particular gene. So you can understand one-to-many interactions. What are the genes that are present within the nucleus, with what kind of genes it is interacting within the nucleus. And then Hi-C is a method by which one can actually, the very high throughput next generation sequencing based technology, which can give you the information about entire genome architecture, where how that each and every chromosome territory is placed within that particular cell type, how that individual chromosomes are organized, how that individual genes are organized. All this information can be gathered from this particular technical Hi-C. So over the in this upcoming 2 to 3 slides, I'll be explaining briefly about these methods, because these importance are very powerful. Now with these techniques, now our understanding about genome organization, gene expression has gone to the very next level. So in this 3C technology, for example, think that you have a gene and a regulatory element. We know that when the gene is on, these regulatory elements come to near the gene promoter. So if you have these kind of structures which are mediated through protein-protein interactions, so in this particular procedure, the cells are cross-linked with the formaldehyde, so that all the distally placed or proximally placed DNA elements through protein-protein interactions are cross-linked. This is followed by restriction enzyme digestion, so that they precisely can cut the DNA which leave the coincidence, and then this is followed by ligation. So these remotely placed DNA elements, because of this procedure, because they're proximal within the nucleus, so they can be when they're subjected to ligation, these DNA elements can be ligated. And if you have a primer from promoter region, if you have a promoter from enhancer region, if you perform a polymerase chain reaction, the presence or absence of a PCR product is indicative of how close they are within the nucleus. So indirectly, by using this molecular tool, one can tell which gene, which part of the gene is close to what kind of enhancer or what kind of regulatory elements. Similarly, as I said, the 4C technology by using this kind of 3C technology, scientists have identified the putative enhancers for a given gene. Scientists also identified the interaction between centromeres for chromosomes in case of yeast. As I said, 4C is a technology by which one can identify gene-gene interactions within the nucleus. I will come to this particular why that genes will interact within the nucleus. And then as other most powerful technology called Hi-C. Hi-C is similar to 3C, where cells are cross-linked with formaldehyde. They are digested with Hindley restriction enzyme. It leaves this kind of coincidence. This is followed by end filling with biotin nucleotides, followed by ligation with T4 DNA ligase. And then later, after decross-linking, you see that all these ligated junctions are having biotin nucleotide. So after sonication, and then one can selectively enrich these junctions with the help of a paramagnetic beads coated with the streptavidin, and then these millions of chimeric DNA fragments which are enriched by this particular phenomenon can be subjected to Illumina high throughput sequencing, and it will give you pairwise interactions, which is the X region interacting with the other Y region. So you have a complex network of interaction matrix, which can be used to deduce this kind of matrix. For example, Hi-C, if you perform, the whole you see that whole genome matrix can be derived from the data, and this data is very much helpful to understand which chromosome territory is placed where within the context of a nucleus, chromosome territory positioning, and the neighborhood can be derived. Further, if you look into individual chromosome, you can zoom into individual chromosomes, and then you can see that a particular chromosome specific interaction matrix that will help us to deduce how that individual chromosome territory is organized within the nucleus, how that euchromatin, heterochromatin is placed with respect to each other. Further, deep into individual domains, as you can see here, which can be zoomed into, it can reveal this kind of a triangular box-like structure, which actually is nothing but the DNA loop-like organization seen within the nucleus. This kind of functional loop-like organization can be seen as a self-interacting domain. These are called topologically associated domains. So we know that nucleosomes represent the structural unit of chromatin. These TATs, topologically associated domains, represent the functional unit of eukaryotic genomes. See, now based on this kind of 3C, 4C, IC technology, now we know that the so-called double helix DNA, genetic material, and then again with the help of histones forms nucleosomes, which represent the structural unit of chromatin, which hierarchically folded and form this kind of two domains, A domain and then B domain. B domain is nothing but heterochromatin, which is seen as the nuclear envelope. A domain is nothing but it's the euchromatin, which is towards the nuclear interior. And now this is what exactly the picture you can see from each and every chromosome territory. And then at much higher level, you can see that entire genome. You have a chromosome territories, you have a euchromatin, heterochromatin within the chromosome territory, and then genes are specifically placed either in the euchromatin or heterochromatin, thus establishing a cell type specific genome architecture, which actually decides which gene can be transcribed in a cell type specific manner. Okay, as I showed that this loop-like organization is very, very important. So loop-like organization will ensure that we have a spatio-temporal expression of genes. For example, in one of example, we have 4 loops. The experiment will give you this kind of triangular interaction matrix. You can see that there is a 4 triangles, A, 2A, 2B, and 3. Okay, that is nothing but loop 1, loop 2A, loop 2B, and loop 3. As you can see that in loop 2A, you have a gene, green color. You have a regulatory element in red color. So this particular gene present in loop 2A is transcriptionally active because enhancer is present in the same kind of loop. Similarly, in the like loop 2B, you have a gene that is not transcribed because that gene cannot access the enhancer because it is present in a different loop. So in some disease states, those borders which are present between loop 2A and 2B are erased, thus it will become a single loop. Now we can see that the gene which otherwise silent, now that it can access the enhancer present in a single loop. That is one of the cause for the activation of oncogenes. So our oncogenes are kept in silent in our cells because of this, because enhancers are not active, because of any kind of mutations by various factors which disturb this kind of higher order chromatin organization, loop organization. So disturbance in the loop organization leads to inappropriate accessibility of enhancer elements to the genes. Thus, genes which are supposed to be silent, they can be reactivated. One of the example could be oncogene, thus converting a normal cell to a cancerous cell. That means chromatin higher order organization is one of the fundamental mechanisms by which genes are specifically tightly regulated. Any disturbances to this particular higher order chromatin organization leads to inappropriate expression of genes. That is what exactly manifests as a disease states. Okay, so further, just to give you that, that means genome architecture is very unique for every cell type. Although fact is that every cell has a identical DNA, but the way each and every chromosome is organized within the nucleus is very unique. So for example, liver cell give rise to only liver cell. skin cell by means of mitosis give rise to only skin cell. How that these cells know that they belongs to liver cell or skin cell? They have to transcribe only liver specific genes. Here comes during this division, for example, think that you have a parental liver cell with some kind of a genome architecture which is specific for liver cell. When this particular cell undergoes mitotic cell division, so those individual chromosome territories are condensed to become a chromosomes. Within the chromosome, still you can see that heterochromatin marks and euchromatin marks, blue and orange color, they are retained within the chromosomes in the form of histone modifications, and then after metaphase and then anaphase, these individual chromosomes are segregated to.
చెప్పండి
Sorry?
మెసేజ్ చెప్పండి ఇది ఎందుకంటే ఇక్కడ ఇస్తాం కదా మెయిల్ అడ్రస్ చెప్పండి
Yeah. Okay. So and then at the telophase stage, the now cytokinesis carried by cytokinesis and then karyokinesis leads to the formation of two daughter cells. As you can see that in these daughter cells, the genome architecture of a parental cell are still memorized through mitotic cell process, and the same daughter cells also acquire the same kind of genome architecture. So the genome architecture can be maintained in the daughter cells. That means they also memorize the gene expression pattern. Thus, the liver cell give rise to only liver cells, and then skin parental cell give rise to only skin parental daughter cells, because they maintain the genome architecture specific to the particular cell type. Hence, they maintain the genome architecture as well as the gene expression patterns. And then now we know that during cell differentiation, for example, you have an embryonic stem cell, which eventually can give rise to a neural progenitor cell. Neural progenitor cell eventually give rise to astrocytes, for example. If you look into this particular genome in cell differentiation process, what happens to heterochromatin and then euchromatin dynamics? So by using all these technologies, scientists have learned that, for example, in embryonic stem cells, all embryonic stem cell specific genes are transcribed. They will be in the loop organization. All those genes which are not transcribed in embryonic stem cell, they are seen at the nuclear periphery, and they are heterochromatinized, and they are part of the nuclear lamina. Same cell, when you differentiate them to undergo a neural progenitor cell, all those neuron specific genes are now looped away from the nuclear lamina, whereas embryonic stem cell specific genes are now become the part of the lamina. Thus, they change the genome architecture specific for neural progenitor cell. Again, when these cells are undergoing differentiation to other cell types such as astrocytes, you see that accordingly new selective genes are relocated either from heterochromatin to euchromatin or euchromatin to heterochromatin. That means genes dynamically shift their location from nuclear periphery to interior or interior to nuclear periphery. That is what exactly the transcription on-off state of genes which are reminiscent for a particular each and every cell type. Okay, so all these slides I try to explain you the structural organization of genome, where we learned that although all cells are having identical DNA sequence, but each and every chromosomal DNA is uniquely placed within the nucleus, and then their positioning and then neighborhood or cell type specific. That is what exactly decides what kind of genes can be transcribed. So at the same time, now we know that this within the nucleus there are most important nuclear processes can happen. That is one of that nuclear process is transcription. The transcription is the process by which DNA is converted into RNA, and also splicing, mRNA export. So all these things are happening within the nucleus. Although this nucleus are physically having 30,000 genes, if it is human genes, only few genes are transcribed. As I said, around 4,000 to 6,000 genes are transcribed in a given cell type. But when this transcription process, the enzyme responsible for transcription process is RNA polymerase II. So whenever you try to locate RNA polymerase II, as shown here in the red in the picture left, this is the location of a RNA polymerase II enzyme within the nucleus. It always forms this kind of nuclear foci. They're called nuclear domains. And then there is one more protein called CTCF. If you try to locate even this particular protein, it is seen in the form of this kind of a large nuclear foci. So it was puzzled scientists, what are these like this kind of nuclear foci representing RNA pol 2 and then CTCF. For that matter, you take any transcription factor, try to locate them within the nucleus, it always forms this kind of nuclear foci. So from long time, scientists were really puzzled why that these nuclear proteins appear like a nuclear foci. What are they? Why that you at a particular location you find group of proteins aggregated together? What are they? Are they storage sites within the nucleus? Are they is it an experimental artifact? Are they representing any kind of function? So research from the past decade by using this sophisticated 3C, 4C and high-C technology has revealed now what are these important nuclear domains within the nucleus. So there are multiple domains can be seen in the nucleus. For example, polycomb group of protein nuclear bodies, centromere clusters, Cajal bodies, PML bodies, splicing speckles, RNA polymerase II nuclear foci. So there are multiple proteins form multiple domains within nucleus. So it was a long-standing question: what are these nuclear domains? Why that proteins form these kind of domains within the nucleus? Now in biology, these observation and now the kind of experiments that were done in the past 10 years now has uncovered what are these domains, what actually they represent. So before going into details about what are these nuclear domains, I just wanted to tell you that. So soon after the sequencing of a human genome, when scientists learned that our human genome codes for 30,000 genes. But when they try to see the location of these 30,000 genes along these 23 chromosomes of human, they learned that they're not randomly placed here and there. They learned that they're clustered at the primary sequence level. You see that genes are clustered and then you will find the empty space. Again, group of genes are clustered, again you will find the empty space. That means genes are, to start with at the primary sequence level, they are not randomly placed, they are clustered. They're not only clustered, they're co-expressed. Think that among these clusters of genes, if any gene in that cluster is transcribed in a particular cell type, it's most likely that all this group of genes in that particular cluster are transcribed in a cell type specific manner. That means genes at the primary sequence level are not only clustered, but co-expressed in a cell type specific manner. These kind of clustered and co-expressed genes at the DNA are called ridges, regions of increased gene expression. These are very common in human genome. Not only human genome, for that matter, each and every organism, you will have the genes are clustered and then co-expressed in a cell type specific manner. Okay, the other thing that what I'm trying to say is that, so when scientists try to locate RNA polymerase II enzyme within the nucleus, so expected thing by means of immunofluorescence, where one can use RNA polymerase II specific antibodies, one can detect their color by using some kind of fluorophore. So here, based on our theoretical expectation, if I try to perform an immunofluorescence technique with RNA polymerase II antibodies in a particular cell, I would expect to see this kind of grainy pattern, because although every cell has 30,000 genes in human cells, but we know that 4 to 6,000 genes are transcriptionally active in a given cell type. If 4 to 6,000 genes are transcriptionally active, each and every gene requires RNA polymerase II. So if I try to locate RNA polymerase II enzyme, I would expect to see the at least 4 to 6,000 grainy spots over the nucleus, which are dispersed across the nucleus. But surprisingly, in reality, when scientists have performed RNA polymerase II immunofluorescence, they detected this kind of large nuclear foci. As I shown you in the beginning, also the photographs. So this has really puzzled scientists. Why that transcription, protein involved in transcription, instead of appearing like a diffuse pattern, why that it forms such a large nuclear foci? So this, as I said, by using some of the molecular tools that I mentioned, scientists now unravel what are these actually RNA polymerase II nuclear foci. For that matter, all other nuclear domains that what we know, now we have a explanation. So this is what exactly RNA polymerase II, when you try to detect with kind of red fluorescence, appears like a large nuclear foci within the nucleus. So here in this experiment, they wanted to see this RNA polymerase II concentrated nuclear foci, whether they are storage sites or do they have any kind of transcriptional activity. So for that, in order to locate the this RNA polymerase II nuclear foci, they performed a RNA immunophage technique. So is the RNA, so here they detected RNA polymerase II protein by immunofluorescence, by red fluorescence, and then transcripts for a particular gene, which is known to be transcribed in a cell type, is called globin RNA, by means of a RNA fluorescence in situ hybridization. As you can see, red color representing the RNA polymerase II enzyme in the form of a large nuclear foci. Wherever then when you detect a primary transcript, that is a product of a transcription, RNA, in the form of a green color, you see that always it is seen on the surface of this nuclear foci, suggesting that RNA polymerase II enriched nuclear foci are not the storage site. There is active transcription is going on within this highly aggregated RNA polymerase II locations. So what are these foci? So this, now we know that these are called transcription factories. These are not transcription factors. These centers are called transcription factories. Other name is transcriptional condensates, which has a 1,000-fold higher concentration of RNA polymerase II. And then per cell, you see around 200 to 400 foci can be seen. But if these foci are the sites of transcription, then we know that approximately in a particular cell type, around 8,000 expressed genes are present. That means most likely that each and every nuclear RNA polymerase foci must be harboring multiple gene transcription. Is that true? So in fact, we tested when I was working with Peter Fraser group in Abraham Institute, where we have tested that, for example, you see that in mouse chromosome number 7, there is a gene called globin gene, which is red color, labeled with red color. And there are a lot other genes, ERAF, EUROS, and IGF2, KCN, Q1. These are genes which are present megabases away from hemoglobin gene. Okay, so when you take HBB gene labeled with the red color, and you take ERAF gene labeled with green color, and hide simultaneously together to the nucleus, as you can see in the first panel, where the HBB globin gene is in red color and then ERAF gene is in green color. Both of them are physically separated by 30-40 megabases away from each other. When you try to detect these genes in the nucleus, you see that they are together. So red color you see, and then green color, they are together. That means they give yellow fluorescence in 42% of cell types. Similarly, in other experiment, same globin gene in red color, EUROS, which is present here, labeled with green color, when you try to detect them in the nucleus, again they are seen in 41% of the cell seem to be together. Then when they are together, they are always seen at the nuclear foci, suggesting that genes from same chromosome during transcriptional activation, okay, they come together for transcription process. Not only in same chromosome, even from different chromosome. For example, you see that there are two genes, TFF1 and then GREB1, these two genes, which are transcriptionally active. In absence of estrogen, they are not expressed, they are not transcribed. When they are not expressed, you see that red dots and then green dots represent these two genes, they are away from each other. But when you add estrogen, you see that these two genes which are separated by megabases away, they come together. You see that red color dot and then green color dot, they come together upon estrogen induction, suggesting that for gene activation, genes can come together from very long distance. So this is what exactly the current understanding about how gene transcriptions are orchestrated within the eukaryotic cell nucleus. As I told you that RNA polymerase II nuclear foci, and we have a chromosome territory. As you can see that here, this one particular nuclear foci is enlarged here. The RNA polymerase II nuclear foci is nothing but these are the centers where genes from same chromosome territory or different chromosome territory three-dimensionally cluster for a dedicated transcription control. That means when the genes are transcribed, like unlike our textbook perspective, when the genes transcribe, RNA polymerase moves along the DNA. That is not true here. Here, the group of genes which requires to be transcribed, they three-dimensionally cluster, and then along with RNA polymerase II and then other associated factors, forms a dedicated centers of transcription factors for transcriptional activation. It is like in a hostel. Hostel has only common kitchen. Instead of having a kitchen in each and every room, we have a common kitchen that is more economical perspective, so that each and everyone can go there and then have their meal. Similarly, within the eukaryotic cell nucleus, all genes which requires to be transcribed, they come together within the chromosome or between chromosomes and forms a dedicated centers of transcription called transcription factories or transcriptional condensates. So this is not only for the active genes, even silent genes from different chromosomes. they cluster together in the form of a silencing factories for a dedicated coordinated control of gene silencing. See, this is here exactly I showed, for example, our test perspective where you have a DNA molecule, RNA polymerase sitting, and then when it moves, here the DNA is stable, RNA polymerase will be moves. When they move, and then it forms a corresponding transcript. But in reality, this is not correct. Within the nucleus, the DNA, for example, RNA polymer 2 is more stable, and then DNA is reeled through this particular transcript centers for thus will have a RNA being transcribed. So it is the DNA that moves rather than DNA polymerases. That is what exactly the RNA polymerase 2 nuclear foci experiments suggest. As I said, not only the active genes cluster, even the silent genes. For one example here I'm showing you. In Drosophila, on chromosome number 3R, there is gene called antennapedia. It is called ANTP. Here they label with red color. And there is one more gene which is away from megabases away from this particular gene called ABDB gene, which is labeled with say pink color. So when they try to detect these two genes in a head region of Drosophila, where these two genes are transcriptionally not expressed. When they're not expressed, you see one gene is red color, other gene is orange color, and then there is a protein called polycomb group of protein, which is actually gene required for gene silencing. So these also form some kind of nuclear foci. If you see these two genes are transcriptionally off in the head region of Drosophila, but when you try to see all together, both red spot, orange spot, and then green spot, they are together. What it suggests that genes during silencing, from long distance, they come together along with these factors called polycomb group protein for gene silencing. Look at the same thing when in the posterior part of Drosophila, one of the genes, that is ANTP1, is transcriptionally active, whereas ABD gene is transcriptionally active, ANTP gene is repressed. You can see the repressed allele is within the polycomb group of protein, where the active allele moves away from the silencing factors. Thus, both for gene activity and gene silencing, genes cluster from same chromosome or interchromosomally for either for activation or silence. So this is a model to show that genes are repressed. Transcriptionally, they're clustered, but when one is active, it moves away from the silencing factor. So now, just to give you an overall view of the perspective of the genome organization, and then it's linked with the gene expression. As you can see that this electron micrograph picture clearly shows the organization of a nucleus. With a nucleus, you find electron dense heterochromatin and euchromatin. And this heterochromatin, as you can see, it is bound with a protein called lamins. And then if you try to see the individual chromosome territories, you see that all the within the chromosomal territory or chromosomal DNA for that matter, all the regions which are silenced, they have this kind of lamina binding proteins, and these proteins are clustered on the linear scale, and then they're pushed towards the nuclear periphery, and then they form the heterochromatin. All those regions within the linear DNA which are having activation modification, which the genes are active, they're highly decondensed, and they are seen towards the nuclear interior. If the gene is present in the nuclear interior, the most likely that is transcriptionally active. The gene is present within the nuclear lamina or the green colored structure or heterochromatin, the gene is most likely to be transcriptionally silent. Okay, so then in that context, then you must be really wondering, we know that these kind of like all cells do not transcribe all the genes. That means every cell has a unique genome architecture, unique chromosome territory. So genes are not are somehow switched between heterochromatin, euchromatin, euchromatin, heterochromatin. That is what exactly happens during cell differentiation. For example, in this example, a stem cell is undergoing differentiation process, coming to an intermediate stage, and the intermediate state finally it will be differentiated to a differentiated cell. So if you look into this chromosome territories of a stem cell, chromosome territories are highly randomly placed within the stem cell. They're highly mobile. But once it is determined to become intermediate cell, now it becomes, you see that chromosome territories are non-random, and in a different cell, you have a unique positioning of each and every chromosome territories. So stem cell differentiation into a differentiated cell is accompanied by random organization of chromosome territories to non-random organization of chromosome territories in a differentiated cell. If it, so as I said, these kind of nuclear domains that what you see, which is like RNA polymerase nuclear foci, are formed by a phenomenon called liquid-liquid phase separation. Now, the entire biology, wherever you see any kind of nuclear functions mediated through any kind of nuclear proteins, they all perform this kind of a phenomenon called liquid-liquid phase separation. It is just like if you have a beaker of water, if you try to add a oil droplet into the beaker and then try to shake it, you have a pure emulsion, right? So oil also is a liquid, and then water also is liquid. So within the liquid, over a period of time, if you try to keep this kind of a beaker with oil droplet, over a period of time, all the oil droplets will try to fuse together on some single oil droplet. Similarly, within the nucleus, the proteins with similar kind of functions, genes with similar kind of function, with same kind of associated factors, they over a period of time, they cluster together by using this kind of physicochemical phenomenon called liquid-liquid phase separation. Thus, it forms nuclear domains, and then which are actually active nuclear processes are happening in these particular structures. So here I almost covered both structural as well as the functional aspect of eukaryotic genomes. How that although all cells are having identical genomes, how that epigenetic mechanisms will influence the cell type specific gene expression program. So just to give you some more basic information, see one of the very fundamental questions is that you see our genomes, if you see, we have 23 pairs of chromosomes. That means 46 chromosomes are there in each and every nucleus. And each and every chromosome has a DNA. If you try to put together all these DNA molecules together, 46 chromosomes, it measures around 2 meters. It will be really puzzling to learn that each and every cell we have nearly 2 meters length of DNA, which is packaged in almost 10 micrometer cell nuclear value. So that suggests that you have a long polymer of DNA. How to keep such a long polymer of DNA in the smallest possible volume? For example, think that if you have 2 meter length of thread, and if I ask you to put them in a smallest possible volume, then all of us will come to this kind of organization, right? Just take the thread and then try to put it in a smallest possible volume. Then it occupies the least possible volume. But disadvantage with this kind of organization is that if you want to access the start point of this particular thread, it is almost impossible. Even if you want to access any other region, it's almost impossible. Certainly, this is not the way our genomes are packaged within the nucleus. Then how our genomes are packaged? We learned about chromosome territories. So now in polymer physics, we have this kind of organization. For example, you have certain length of DNA polymer, but if you want to arrange them in a particular volume, you see in these two pictures, the volume is same, the length of the DNA is same, but you see the way this DNA is placed within this particular volume. Here you see that in the first picture, the Pinot curve pathway, it is called. First, the start point is here, red color. The end point is blue, it is here. If you see, in a given, the nearest fragment is always near to the start point, okay, in this organization. Whereas here, in the Hamilton pathway, you see that start point is here, the end point is somewhere here, but you see the way it is arranged, very randomly arranged. So you see that this is the start point. This is somewhere in the middle. It is still seen at the near to the start point. But here, the somewhere which in the middle can be never seen at the start point. So this kind of organization will explain how the DNA within the nucleus is actually organized, suggesting that this kind of Pinot curve pathway is the one by which our genome is actually organized. You see here, but this is the linear scale, two-dimensional scale. Put it in the three-dimensional scale, it will tell you that DNA is a fractal geometrically organized. This kind of organization, where you see that a structure is organized following the Pinot curve pathway, is called fractal geometry, which is not free. That will explain why the DNA fragments will never form not within the nucleus. So what is this fractal geometry? As I said that DNA within this nucleus is organized in the form of fractal geometry. What is this fractal geometry? So fractal geometry is not a very new thing. It's very common. You see almost, you see this kind of, for example, in this example, you see that a fractal is a geometric pattern that is self-similar, meaning that the whole pattern is constrained within its parts. In other words, if you zoom into any part of the pattern, it looks the same. Similar patterns reoccur at progressively smaller scales. See, at the higher order scale, This is the leaf organization. If you come down to other level, you have the same kind of structure as seen here. And then if you go down at the tertiary level, you'll find the same kind of structure. If you go down, if you can use a microscope, you find the same kind of structure repeated at the multiple scales of organization. This kind of organization is called fractal geometry. This is not only the case. For example, here in this example, you see the kind of structure you see at the macroscopic level, the same structure repeated at the secondary level. Again, in the tertiary level, same structure again repeated at the third level. So this kind of organization will ensure that you have a large volume of things are packed within the smallest volume. Still, each and every part is accessible to any kind of nuclear proteins. For that matter, our universe itself is a fractal, right? So from that, you can understand that genome within the nucleus, 2 meter length of genome within the nucleus, with the help of a fractal geometric organization, is organized. Thus, it is not free, and then it fulfills all the nuclear functions required for where each and every part of the chromatin is highly accessible. So it is the same. So genomes, if you go to individual chromosome territories, within the chromosome you have euchromatin, heterochromatin. Again, same kind of structures are seen at the down to the individual genes. Okay, so I think so. With that, you can understand that although every cell has a 2 meter length of DNA, they never form knots because they are organized in very sophisticated form in the form of chromosome territories, and then euchromatin, heterochromatin, and then again in the form of a topological or short domains, and in the form of a DNA molecules. Okay, so other important aspect which I wanted to touch upon is that gene positioning and transcription. So from these, whatever we discussed so far, we learned that gene positioning really matters. For example, if any gene is active, that means 2 copies because we are diploid animals. If you take any cell, we have 2 copies for a given gene. One is from mother gene, other is from father genome. So there is technique called DNA RNA fish. By using DNA fish, you can detect a particular gene within the nucleus. By means of RNA fish, you can detect whether the gene is transcribed or not. So if simultaneously do both the things at a time, DNA RNA fish will not only detect the gene positioning but also detect its transcription status. So here DNA is detected with red color, RNA is detected with green color. For active gene, you can see that both are seen at the nuclear interior, 2 dots seen at the nuclear interior, and both of them are emitting green color. That means gene is transcriptionally active. If you take a silent gene in a given cell type, you see that you perform DNA fish, red color, it is showing nuclear periphery. If you perform the RNA fish, you don't detect RNA green color because they're not transcribed. We have some kind of genes in our cells that are called imprinted genes. That means their transcription is parent of origin specific manner. If a copy from mother may be silent, copy from father may be transcriptionally active. If you see such kind of imprinted genes, and then try to do DNA RNA fish for that particular gene, you see that DNA you can detect 2 spots. One is at nuclear periphery, one is at nuclear interior. And then when you perform RNA fish for the same gene, you see that for these genes, the gene that is present at the nuclear interior is giving green color, that is transcriptionally active. The copy that is seen in the nuclear periphery is not giving green color because it is not transcriptionally active. That means gene positioning dictates cell-specific, the gene-specific transcription program. Further, during replication, because during replication, what happens is that entire genome has to be duplicated. And then during this replication, some genes they transcribe, some genes they replicate early, some genes they replicate late. So it was learned that genes that replicate early are nothing but those genes which are transcriptionally active in a given cell type, they tend to replicate early. All those genes which are silent in that particular cell type, they tend to replicate late. So this replication timing can act like an epigenetic memory. Thus, genes are selectively either switched on, switched off. The gene is replicating early. It's most likely that when it comes to the daughter cell stage, that gene is transcriptionally active. The gene replicates late. It's most likely that that gene transcription is silent in the daughter cells. Okay, so with this, now we can appreciate that although all cells of our body are having identical genome, but the way that each and every piece of DNA is placed three-dimensionally will actually is unique for each and every cell type. This is not the only epigenetic mechanism by which genes are regulated. So our genes are heavily regulated at multiple levels. One is pre-transcription level. That means before the cell is being formed, its transcription is decided. What kind of genes can be transcriptionally active? So that is because of DNA methylation, replication timing, chromatin accessibility, chromatin condensation, chromatin supercoiling, and then three-dimensional organization of chromosome before transcription. So they play an important role they will decide which genes are going to be transcriptionally active or silent. And then genes also can be regulated at the transcription level, that is at the level of promoter proximal pausing, presence or absence of a specific cell type, specific transcription factors, histone modifications, and then chromatin remodeling. They play an important role for regulated transcription at the transcription level. And then genes also regulated at post-transcription level. So that means they are transcribed, but after transcription, by means of alternative splicing, and then microRNA, long non-coding RNA, circular RNA, and then RNA modifications, RNA binding proteins, mRNA stability and degradation acts like other important key epigenetic mechanisms by which genes are regulated post-transcriptionally. Further, genes are also regulated translation. That means genes are transcribed. They're not necessarily that all of them are translated. That means they are regulated at even a translation level. So there are specialized ribosomes, initiation of translation, elongation, termination, and then microproteins. They play an important role in controlling the gene expression at the translation level. Even at the post-transcription translation level, genes are regulated in terms of protein post-translation modifications, protein stability, and then protein degradation. From this, you understand that in eukaryotes, being such a complex multicellular organisms, their genomes are evolved, and then their gene expression pattern tightly controlled by multiple mechanisms of pre-transcriptional, transcriptional, post-transcriptional, translational, and then post-translational mechanism that will ensure that no inappropriate genes are transcribed in inappropriate cells. So each and every cell transcribes a unique set of cell type specific genes only. Any perturbations in all these epigenetic mechanisms can lead to disease state. You take any disease state is nothing but is a dysregulation of gene expression can be mediated through any of these particular disturbances to any kind of these epigenetic processes. So with this, I will stop here. If you have any questions, I will take questions. Thank you very much. So any questions, please.
Hello, sir.
Yes, please.
Good afternoon, sir.
Yeah.
So your talk was really enlightening and gave me to know about very new technique. So as we all know, like all the genes are present in all the cells, and you have emphasized that the positioning of the chromosomes inside the nucleus is responsible for the expression, specific expression and the specificity of the cell.
Yes.
Now my doubt is, if I pluck out the nucleus from suppose say a renal cell and put it in a liver cell,
Yeah.
what will happen to that cell? What will happen?
So, can you repeat the question? So, if you take the nucleus from one cell and then put it back into other cell type, you're asking, right?
Yes, yes. Sir.
Yeah, that is a very nice question. For example, you see, cytoplasm has some kind of protein factors. Nucleus has some kind of factors, right? So, if I take for example, I take a liver cell, I will remove the nucleus, and then I will take a nucleus from neuron, I will put it into the liver cytoplasmic environment. So in this context, so you have it is at the end it will be it is neither a liver cell nor a neuronal cell. It will have a different set of gene expression pattern. So, for example, in this example, maybe I can tell you, if you take any kind of, for example, differentiated somatic cell, if you add these three factors like SOX2, NANOG, and OCT4, these are called MNOC factors, you can convert a differentiated cell back to embryonic stem cell like cells. This is called induced pluripotency. There, what happens is that every cell has a defined epigenome, right? So they extensively reprogram and then forms this kind of chromatin, which is reminiscent of a stem cells.
That is being their stem, like differentiating the stem cell. But I am saying completely differentiated cell, nucleus exchange.
Yeah, differentiated cell, as I said, any differentiated cell, if you put it in different environment, presence of particular factors in that particular environment will reset that complete gene expression patterns in that particular nucleus.
The floor is open for discussion from any NRC.
Hello, good afternoon, sir.
Yes, please.
Myself, Dr. Lalita Kumari from NRC Nellore, sir. Is there any specific function for the silent genes or imprinted genes, sir?
Silent genes are?
Imprinted genes.
Oh, yeah, see.
Imprinted genes.
Yeah. See, for example, if you take any particular cell type, some genes are transcriptionally active. They're called active genes, and some genes are silent, right? They're called silent genes. Among these genes which are active, they're active when they transcribe from both the copies. Like every cell we have maternal copy as well as paternal copy, right? So when they transcribe from both the copies. But there are some kind of genes, they're called imprinted genes. These imprinted genes, out of the two copies, only one copy is transcriptionally active, other copy is transcriptionally silent. The copy that is silent is seen as the nuclear periphery. The copy that is active is seen as the nuclear interior. These kind of genes are called imprinted genes. In humans, we have nearly 75 genes which undergo imprinted pattern of gene expression.
Thank you, sir.
Or to PDK and college school. Raise their hand.
Sir.
Yes, please.
PDK and NRC. Sir,
Yes.
why do we chromatic repositioning grow in some regions of the nucleus and less in some regions? Why? What are the reasons?
Actually, your voice is not clear to me. I can't hear. If someone can.
Chromatic repositioning grow in some regions of the nucleus and in some regions it is less. Why?
Excuse me. Chandramodi sir. Please check your mic and audio.
Chromatic repositioning more in some regions of the nucleus. Sir, why?
The voice is not clear, sir, from your end. Chandramodi sir, PDK and Chittur.
Chromatic repositioning occupation there nucleus is not true for vice. Is not true. Chromatic repositioning.
Sorry, sir. Actually, I think somehow we can't hear you.
Excuse me, sir.
Yes, please.
Sir, you have told that 6 billion base pairs of DNA are present, actively present. So during its expression in the course of life, how many billion base pairs are actively present during the life?
Okay, if you take a human genome, the percentage of genome which codes for gene is 33%. Okay, among these 33%, if you look at the exonic portion as well as intronic portion, all the exonic portion of all the transcribed genes represents less than 1% of the human genome. So the actual percentage of the human genome which transcribes and then codes for protein is less than 1%. So from that you can imagine we have 6 billion base pairs of nucleotides. From that means only 1% of that will be transcriptionally active. Rest all other DNA, although they call it a junk DNA. Now we know that this DNA is not a junk. It is actually play an important regulator for spatio-temporal expression of genes in different cell types during different developmental contexts.
Any other faculty from other NRCs? Or else over to GDC Tanuku.
Yes, please.
Thank you, sir.
Proposal of board of NRC Tanuku.
That's part, sir. Myself and Ramakrishna, Srinivasa, sir, from University of Hyderabad. Sir, you are inspired to cover the related.
Sorry to interrupt you, sir. Ramakrishna, sir. Audio is not clear, sir. Please check once.
Sir, what now? Now it's okay, sir. Sir. Sir, development of identity, multi-protein cells, one gene regulated genetic information in the form of DNA is stored in the hierarchical organization of chromatin, genome, fragmentation and classification including actual, repressive, DNA replication, and physical effects of replicated chromosomes, DNA structure and its regulated genome, layers of chromatin, genetic modification during the nuclear genome, segregated repressive compartments. Chromosomal DNA is constrained replication in the spectrum candidates like all given by especially related to so very much thankful to use that to market.
Srinivasa, sir.
Yes, please.
Commissioner of College Education and all the NRCs across the Andhra Pradesh. We profusely thank you, sir. You have given an elaborate and marathon, wonderful, extensive talk on recent developments and recent research activities regarding the technologies also. And it is a very wonderful, informative, scientific talk, sir. Thank you so much. On behalf of all the participants, sir, thank you, sir. Thank you so much.
Thank you so much for the opportunity. Thank you very much. It is nice interacting with you.
You can sign off, sir. Thank you, sir.
Thank you very much. Bye.
Now the participants can leave for tea.